Hello, science enthusiasts, curious minds, and fellow travelers on this incredible journey of discovery. Welcome back to Vibing Science. Tonight, we embark on an ambitious voyage, a deep dive into the very essence of existence itself, biology. From the microscopic architecture of a single cell to the sprawling interconnectedness of global ecosystems, we'll unravel the intricate mechanisms that allow life to flourish. If you find yourself captivated, a like, a comment, or a share truly helps our channel grow and reach more inquisitive spirits. Let's begin. From the quiet dance of stardust in the cosmic dark to the vibrant, bustling complexity of a single living cell, we embark on a journey. A journey into the very essence of existence, into the magnificent, intricate science of life itself. Tonight, we delve into biology, the profound scientific study of all that lives and breathes and grows. It is a vast tapestry, weaving together countless threads of understanding, from the unseen molecular machines within us to the sprawling ecosystems that blanket our world. At its core, biology is driven by 5 fundamental themes, pillars upon which our understanding of life rests. First, the cell, the basic, irreducible unit of all life, a miniature universe of activity. Second, genes and heredity, the silent architects of inheritance, passing blueprints from one generation to the next. Third, evolution, the relentless sculptor of biological diversity, ever shaping and refining life's forms. Fourth, energy transformation, the vital engine that sustains every living process, from the humblest bacterium to the most complex mammal. And finally, homeostasis, that delicate internal balance, the constant striving for stability amidst a world of flux. To truly grasp the grand scope of life, we must examine it across multiple scales of organization. Imagine zooming in closer and closer from the vast populations that fill our planet, to the intricate communities they form, down to individual organisms, then into their very cells, and finally to the molecules that compose them. Each level reveals new layers of wonder. Within this grand exploration, we find specialized fields, each a window into a particular aspect of life. Molecular biology, peering into the submicroscopic world. Physiology, unraveling the workings of living systems. Ecology, charting the relationships between organisms and their environments. Evolutionary biology, tracing life's epic journey through time. Developmental biology, observing the miraculous transformation from a single cell to a complex being. And systematics, the art and science of classifying life itself. Modern biology, in its deepest sense, is profoundly rooted in 2 revolutionary ideas. The first, of course, is Charles Darwin's theory of evolution by natural selection, a concept that fundamentally reshaped our understanding of life's diversity and its interconnectedness. The second, equally transformative, is our molecular grasp of genes, those tiny instructions encoded within the double helix of DNA. The unveiling of DNA structure, a story in itself, and the subsequent leaps in molecular genetics have utterly remade our understanding of life, pushing the boundaries of what's possible in medicine, agriculture, biotechnology, and even environmental science. Consider this. Life on Earth is believed to have ignited its spark over 3.7 billion years ago. From those primordial beginnings, it has blossomed into an astonishing array of organisms, from the simplest single-celled archaea and bacteria, resilient pioneers of existence, to the magnificent multicellular kingdoms of plants, fungi, and animals, each a testament to life's boundless creativity. Biologists, in their pursuit of order amidst this glorious chaos, meticulously classify organisms based on shared characteristics and their evolutionary kinship, building intricate taxonomic and phylogenetic frameworks that map the branching tree of life. These organisms, in turn, do not exist in isolation. They engage in a ceaseless ballet of interaction with each other and their surroundings within complex ecosystems, playing vital roles in the flow of energy and the intricate cycling of nutrients that sustain the entire planet. Biology is not a static field. It is a living, breathing discipline, constantly incorporating new discoveries and technologies. Each new insight deepens our awe, enhancing our understanding of life's processes, and perhaps most importantly, empowering us to confront some of the most pressing challenges of our time. The relentless march of disease, the existential threat of climate change, and the tragic loss of biodiversity that diminishes us all. Even the very word biology carries a history, a whisper from ancient Greece. It springs from bios, meaning life, and logia, meaning study of. While the compound term made appearances in earlier works, it was in the early 19th century that it truly took root, independently introduced by several brilliant minds like Gottfried Reinhold Treviranus and Jean-Baptiste Lamarck, solidifying its place as the dedicated pursuit of understanding living nature. Our scientific lineage, which intimately includes medicine, stretches back to the ancient civilizations of Egypt and Mesopotamia over 5 millennia ago. Their accumulated wisdom flowed into the philosophical currents of ancient Greece, where figures like Aristotle, a titan of thought, delved deeply into biological causation and the breathtaking diversity of life. His successor, Theophrastus, meticulously laid the groundwork for the scientific study of plants. Later, the scholars of the medieval Islamic world, like Al-Jaiz and Al-Dinawari, expanded these horizons, contributing extensively to botany, anatomy, and physiology, always building upon the intellectual foundations laid by the Greeks. But a truly transformative moment arrived with the dramatic improvement of the microscope by Anton van Leeuwenhoek. Suddenly, the unseen world burst into view. Scholars marveled at spermatozoa, bacteria, infusoria, and an entire hidden realm of microscopic life. These discoveries ignited a fervent new interest in entomology and spurred the development of delicate microscopic dissection and staining techniques. The microscope, a humble lens, profoundly reshaped biological thought, revealing that the very fabric of life was far more intricate than anyone had dared to imagine. By the early 19th century, a revolutionary idea began to crystallize: the central importance of the cell. In 1838, Schleiden and Schwann championed what are now universal principles. First, that the cell is the basic unit of all organisms, and second, that each individual cell possesses all the characteristics of life. Initially, they struggled with the idea that all cells arose from the division of other cells. However, with the groundbreaking work of Robert Remak and Rudolf Virchow, this third tenet was definitively established. By the 1860s, these three principles coalesced into the bedrock of modern biology: cell theory. Simultaneously, the quest to classify and categorize nature became a major focus for natural historians. Carl Linnaeus, in 1735, published a foundational taxonomy for the natural world, and by the 1750s, he had introduced the scientific naming system we still use today for countless species. Other brilliant minds, like Georges-Louis Leclerc, Comte de Buffon, began to challenge prevailing notions, suggesting that species were not fixed entities, but malleable forms, even hinting at the audacious possibility of common descent, a concept that would echo through the coming decades. The stage was being set for one of the most profound intellectual shifts in human history. Serious evolutionary thinking truly began to take shape with Jean-Baptiste Lamarck, who offered a coherent, albeit ultimately incomplete, theory of evolution. But it was the British naturalist Charles Darwin who, through an extraordinary synthesis of diverse observations and insights, forged a truly successful evolutionary theory based on natural selection. He wove together the biogeographical insights of Humboldt, the uniformitarian geology of Lyell, Malthus's challenging writings on population growth, and his own meticulous morphological expertise gained during years of extensive natural observations. Remarkably, Alfred Russel Wallace, working independently, arrived at strikingly similar conclusions, underscoring the power of observation and rational inquiry. Then, a quiet monk in a monastery garden sowed the seeds of yet another revolution. Gregor Mendel, in 1865, laid the foundational principles of biological inheritance, a discovery whose immense significance would remain largely unappreciated until the early 20th century. When his work was finally rediscovered, it provided the missing piece, allowing the reconciliation of Darwinian evolution with classical genetics, giving rise to what we now call the modern synthesis, a unified theory of life's unfolding. The mid-20th century brought another seismic shift. In the 1940s and early 1950s, a series of elegant experiments, notably by Alfred Hershey and Martha Chase, pointed unequivocally to DNA as the carrier of those mysterious trait-carrying units we now knew as genes. And then, in 1953, the iconic discovery of the double helical structure of DNA by James Watson and Francis Crick launched us into the breathtaking era of molecular genetics. From that point onward, biology expanded exponentially into the molecular domain. The genetic code itself was cracked by the combined efforts of Har Gobind Khurana, Robert W. Hawley, and Marshall Warren Ehrenberg, revealing how the sequence of DNA nucleotides translates into the proteins that build and operate life. This molecular revolution culminated, perhaps most grandly, with the launch of the Human Genome Project in 1990, an audacious undertaking to map the entirety of our own genetic blueprint. At its most fundamental, life is chemistry. Every organism, from the smallest microbe to the largest whale, is composed of a specific repertoire of chemical elements. Oxygen, carbon, hydrogen, and nitrogen collectively account for a staggering 96% of the mass of all living things. The remaining, equally crucial, elements include calcium, phosphorus, sulfur, sodium, chlorine, and magnesium. These elements, in their countless combinations, form the compounds that are the very basis of life. Biochemistry, the study of the chemical processes within and related to living organisms, and molecular biology, which seeks to unravel the molecular underpinnings of cellular activity, are the fields that illuminate this intricate chemical dance of life. Consider water. Life, as we know it, arose from Earth's primordial oceans some 3.8 billion years ago, and water remains the most abundant molecule in every organism. Its importance cannot be overstated. Water is an exceptionally effective solvent, capable of dissolving a vast array of solutes, from sodium and chloride ions to complex small molecules, forming the aqueous solutions essential for life's chemical reactions. Its unique molecular structure, a small, polar molecule with a bent shape, where two hydrogen atoms are covalently bonded to one oxygen atom, is the key. Because of the polar OH bonds, the oxygen atom carries a slight negative charge, and the hydrogen atoms a slight positive charge. This polarity allows water molecules to attract each other through hydrogen bonds, giving water its remarkable cohesive properties, leading to phenomena like surface tension. Water is also adhesive, clinging to other polar or charged molecules. One of water's most unique and profoundly important properties is its density anomaly. It is denser as a liquid than it is as a solid or ice. This means ice floats on liquid water, insulating the aquatic life below from freezing temperatures, a crucial factor in the survival of countless species through Earth's colder periods. Furthermore, water possesses a high specific heat capacity, meaning it can absorb a large amount of energy before its temperature significantly increases. This buffers living systems against drastic temperature fluctuations. It also requires a substantial amount of energy to break water's hydrogen bonds and convert liquid water into vapor, a property crucial for evaporative cooling in many organisms. Even at a molecular level, water is not entirely static. Each molecule continuously dissociates into hydrogen and hydroxyl ions before reforming. In pure water, these ions are balanced, resulting in a neutral pH, a delicate equilibrium vital for cellular function. Beyond water, nearly every molecule that makes up an organism contains carbon, making organic compounds, those molecules containing carbon bonded to other elements like hydrogen, utterly central to life. Carbon is a remarkably versatile atom, capable of forming up to 4 covalent bonds with other atoms, enabling it to construct incredibly diverse, large, and complex molecules. Picture a single carbon atom forming 4 single bonds, like in methane, or 2 double bonds, as in carbon dioxide, or even a triple bond, as in carbon monoxide. Moreover, carbon atoms can link together to form incredibly long chains, like octane, or intricate ring-like structures, such as glucose. The simplest organic molecules are hydrocarbons, chains of carbon atoms adorned with hydrogen. But these carbon backbones can be further embellished by other elements like oxygen, phosphorus, and sulfur. These specific groups of atoms, bonded to a central carbon skeleton, are known as functional groups. They're the chemical workhorses that dictate a molecule's behavior. 6 prominent functional groups are omnipresent in living organisms: the amino group, carboxyl group, carbonyl group, hydroxyl group, phosphate group, and sulfhydryl group, each playing distinct roles in the grand choreography of biochemistry. Remarkably, the 1953 Miller-Urey experiment vividly demonstrated that organic compounds could spontaneously arise from inorganic matter under conditions mimicking early Earth, offering a powerful suggestion that the complex organic molecules vital for life might have simply emerged from the non-living world, a fascinating hint at the very origins of abiogenesis. These smaller organic building blocks then assemble into macromolecules, the titans of the molecular world. These large molecules are constructed from smaller repeating subunits called monomers. Sugars, amino acids, and nucleotides are the fundamental monomers that combine to form the major classes of biological macromolecules. Carbohydrates, for example, encompass both individual sugar monomers and their sprawling polymer chains. Lipids, a unique class, are largely nonpolar and hydrophobic, repelling water, and include steroids, phospholipids, and fats, playing crucial roles in energy storage and cell structure. Proteins, arguably the most diverse of all macromolecules, are the tireless workers of the cell. They include enzymes, which catalyze reactions, transport proteins, moving substances across membranes, large signaling molecules, conveying messages, antibodies, defending against invaders, and structural proteins, providing shape and support. The basic building block of a protein is the amino acid, and from a mere 20 distinct amino acids, life constructs an almost infinite variety of proteins. Finally, nucleic acids, DNA and RNA, are polymers of nucleotides. Their function is nothing less than the storage, transmission, and expression of hereditary information, the very code of life itself. All of this intricate chemistry culminates in the cell, the fundamental unit of life. Cell theory, that cornerstone of biology, states 3 profound truths. Cells are the fundamental units of life. All living things are composed of 1 or more cells, and crucially, all cells arise from pre-existing cells through the process of cell division. Most cells are incredibly small, typically ranging from 1 to 100 micrometers in diameter, visible only through the magnifying power of microscopes. Broadly, cells are categorized into 2 main types: eukaryotic cells, which possess a true nucleus and other membrane-bound organelles, and prokaryotic cells, simpler forms without a nucleus, such as bacteria. Prokaryotes are exclusively single-celled organisms, while eukaryotes can be either single-celled or multicellular. In the case of multicellular organisms, every single cell in the entire body can ultimately be traced back to a single, fertilized egg cell, a testament to the incredible power of cellular reproduction and development. Every cell, regardless of its type, is meticulously enclosed within a cell membrane, a delicate yet powerful barrier that separates its internal cytoplasm from the external extracellular space. This membrane is a marvel of engineering, a lipid bilayer composed primarily of phospholipids, with cholesterol molecules interspersed to maintain its fluidity across varying temperatures. This semi-permeable barrier is exquisitely selective, allowing small molecules like oxygen, carbon dioxide, and water to pass through freely, while precisely restricting the movement of larger molecules and charged particles, such as ions. Embedded within this lipid bilayer are myriad membrane proteins, some spanning the entire membrane as transporters, ferrying substances in and out, others loosely attached to the outer surface, acting as enzymes or shaping the cell's form. Cell membranes are not passive boundaries. They are active participants in vital cellular processes such as cell adhesion, the storage of electrical energy, and cell signaling, and they serve as crucial attachment points for extracellular structures like the cell wall in plants, the glycocalyx, and the internal cytoskeleton. Within the cytoplasm of a cell lies a bustling metropolis of biomolecules, including proteins and nucleic acids. Eukaryotic cells, in particular, boast a collection of specialized structures called organelles, many of which are themselves enclosed within their own lipid bilayers or organized into distinct spatial units. The cell nucleus, that magnificent command center, houses the vast majority of the cell's DNA, protecting the genetic blueprint. Mitochondria, often dubbed the powerhouses of the cell, are the tireless generators of adenosine triphosphate, or ADP, the universal energy currency that fuels all cellular processes. Other organelles, such as the endoplasmic reticulum, are intricate networks involved in the synthesis of proteins and lipids, while the Golgi apparatus acts as a sophisticated packaging and shipping center. Biomolecules destined for recycling or breakdown, such as worn-out proteins, are expertly engulfed and processed by lysosomes. Plant cells, those green architects of life, possess additional organelles that distinguish them from animal cells. A rigid cell wall provides structural support, allowing plants to stand tall. Chloroplasts, the miraculous solar panels of the plant world, harvest sunlight energy to produce sugars through photosynthesis. Large central vacuoles provide storage, structural support, and are even involved in reproduction and the breakdown of seeds. Eukaryotic cells also possess a dynamic internal scaffold, the cytoskeleton, composed of microtubules, intermediate filaments, and microfilaments. This intricate network not only provides structural support, but is also intimately involved in the movement of the cell itself and the precise positioning of its organelles. Microtubules are built from tubulin proteins, intermediate filaments from fibrous proteins, and microfilaments from actin molecules that interact with other protein strands, all working in concert to maintain cellular integrity and facilitate movement. All cells, in their continuous dance of existence, require energy to sustain their myriad processes. This grand orchestration of chemical reactions within an organism is known as metabolism. The three primary purposes of metabolism are profoundly simple, yet utterly vital. The conversion of food into energy to power cellular functions. The transformation of this food into the monomer building blocks necessary for growth and repair, and the efficient elimination of metabolic wastes. These enzyme-catalyzed reactions allow organisms to grow, reproduce, maintain their complex structures, and respond to the ever-changing tapestry of their environments. Metabolic reactions can be broadly categorized into two types: catabolic reactions, which involve the breaking down of complex compounds. Think of glucose being broken down into pyruvate during cellular respiration, releasing energy. And anabolic reactions, which are the constructive processes, the building up or synthesis of compounds like proteins, carbohydrates, lipids, and nucleic acids, which typically consume energy. The chemical reactions of metabolism are not isolated events. They are meticulously organized into intricate metabolic pathways, where one chemical is transformed through a series of precise steps into another, with each step gracefully facilitated by a specific enzyme. Enzymes are the unsung heroes of metabolism, the biological catalysts that make life possible. They allow organisms to drive desirable reactions that would otherwise require too much energy or proceed too slowly on their own, by cleverly coupling them to spontaneous reactions that release energy. Enzymes dramatically accelerate reaction rates without being consumed in the process, by lowering the activation energy required to convert reactants into products. They also provide a critical point of control, allowing the rate of metabolic reactions to be finely tuned in response to changes in the cell's environment or to signals received from other cells, maintaining that delicate internal balance we call homeostasis. One of the most fundamental metabolic processes, essential for nearly all life forms, is cellular respiration. This is a complex series of metabolic reactions and processes that take place within cells to convert the chemical energy stored in nutrients into adenosine triphosphate, or ADP, simultaneously releasing waste products. These reactions are primarily catabolic, breaking larger molecules into smaller ones, and, in doing so, liberating energy. Respiration is a primary mechanism by which a cell extracts chemical energy to fuel all of its activities. The overall reaction occurs in a series of biochemical steps, many of which involve redox reactions, the transfer of electrons. While cellular respiration is technically a combustion reaction, it occurs in a slow, exquisitely controlled manner within the cell, a far cry from the explosive combustion we might typically imagine, allowing for the gradual, efficient release of energy. Glucose, in the form of sugar, is the main nutrient utilized by both animal and plant cells in respiration. When oxygen is present, this process is known as aerobic respiration, and it unfolds in 4 distinct stages. The first stage is glycolysis, a metabolic pathway that occurs in the cytoplasm, where glucose is converted into 2 pyruvate molecules, yielding a net production of 2 ADP molecules. Each pyruvate molecule is then oxidized into acetyl-CoA by the pyruvate dehydrogenase complex, a step that also generates NADH and carbon dioxide. The acetyl-CoA then enters the citric acid cycle, also known as the Krebs cycle, which takes place within the mitochondrial matrix. At the conclusion of this cycle, the total yield from a single glucose molecule, or 2 pyruvates, is 6 NADH molecules, 2 FADH2 molecules, and 2 ADP molecules. Finally, we arrive at oxidative phosphorylation, the most prolific ADP-generating stage, which in eukaryotes occurs on the inner mitochondrial membrane, specifically within its folds, known as cristae. Oxidative phosphorylation itself comprises the electron transport chain, a series of 4 protein complexes that meticulously transfer electrons from one complex to another. This electron transfer releases energy from NADH and FADH2, energy that is cleverly coupled to the pumping of protons, hydrogen ions, across the inner mitochondrial membrane, creating a powerful proton motive force. This electrochemical gradient, this potential energy, then drives the enzyme ADP synthase to synthesize a large quantity of ADP by phosphorylating ADP. The journey of these electrons culminates with molecular oxygen serving as the final electron acceptor, forming water. But what if oxygen is absent? In such circumstances, pyruvate takes a different path. It is not transported into the mitochondrion, but remains in the cytoplasm, where it undergoes fermentation. During fermentation, pyruvate is converted into waste products that can then be removed from the cell. The primary purpose of fermentation is to regenerate the electron carriers, specifically oxidizing NADH back to NAD+, so that glycolysis can continue to produce a small amount of ADP even without oxygen. In the absence of oxygen, fermentation prevents the accumulation of NADH in the cytoplasm and ensures a continuous supply of NAD+ for glycolysis. The specific waste product of fermentation varies depending on the organism. In our own skeletal muscles, during strenuous exercise when energy demands outstrip oxygen supply, the waste product is lactic acid in a process known as lactic acid fermentation. Lactate is formed when hydrogen atoms combine with pyruvate, regenerating NAD+. This lactate can then be utilized later, for example, as a precursor for liver glycogen. When oxygen becomes available again during recovery, NAD+ can accept hydrogen from lactate to fuel further ADP production. In yeast, however, the waste products are ethanol and carbon dioxide, a process known as alcoholic or ethanol fermentation, the basis of brewing and baking. The ADP generated in all these fermentation processes is produced via substrate-level phosphorylation, a method that, crucially, does not require oxygen. While cellular respiration extracts energy, photosynthesis builds it. Photosynthesis is the incredible process utilized by plants and other organisms to convert light energy from the sun into chemical energy. This chemical energy is then stored in carbohydrate molecules, such as sugars, which are synthesized from carbon dioxide and water. In most cases, a vital byproduct, oxygen, is released into the atmosphere. Most plants, algae, and cyanobacteria perform photosynthesis, a process that is overwhelmingly responsible for producing and maintaining the oxygen content of Earth's atmosphere, and it supplies the vast majority of the energy necessary for virtually all life on our planet. Photosynthesis also unfolds in distinct stages: light absorption, electron transport, ADP synthesis, and carbon fixation. The initial step, light absorption, occurs when light energy is captured by chlorophyll pigments, complex molecules embedded within proteins in the thylakoid membranes of chloroplasts. This absorbed light energy is then used to remove electrons from a donor molecule, typically water, and transfer them to a primary electron acceptor, a quinone designated as Q. In the second stage, these energized electrons embark on a journey through a series of electron carriers until they reach a final electron acceptor, which is usually the oxidized form of NADP+, reducing it to NADPH. This process occurs within a protein complex called photosystem I-psi. Crucially, the transport of these electrons is coupled to the movement of protons, or hydrogen ions, from the stroma, the fluid-filled space within the chloroplast, into the thylakoid lumen, the space within the thylakoid. This creates a powerful pH gradient across the membrane, with a higher concentration of hydrogen ions in the lumen, analogous to the proton motive force generated across the inner mitochondrial membrane during aerobic respiration. During the third stage of photosynthesis, the potential energy stored in this proton gradient is harnessed. Protons move down their concentration gradient, flowing from the thylakoid lumen back into the stroma, passing through the remarkable enzyme ADP synthase. This movement drives the synthesis of ADP by ADP synthase. The NADPH and ADP generated by these light-dependent reactions, the second and third stages, provide the essential energy and electrons to power the synthesis of glucose. This occurs in a sequence of light-independent, or dark, reactions collectively known as the Calvin cycle, where atmospheric carbon dioxide is fixed into existing organic carbon compounds, such as ribulose bisphosphate, RuBP, ultimately producing sugars that fuel the plant's growth and can be consumed by other organisms. Life isn't just about internal processes, it's also a constant conversation. Cell signaling, or communication, is the fundamental ability of cells to receive, process, and transmit signals within their environment and with each other. These signals can be non-chemical, such as light, electrical impulses, or heat, but more often, they are chemical signals or ligands which interact with specific receptors. These receptors can be found embedded within the cell membrane of another cell or located deep inside a cell. There are generally 4 sophisticated types of chemical signaling. In autocrine signaling, the ligand affects the very same cell that releases it, a cellular monologue. Consider tumor cells, which can reproduce uncontrollably precisely because they release signals that initiate their own self-division. In paracrine signaling, the ligand diffuses to nearby cells, influencing their behavior over short distances. For example, brain cells called neurons release neurotransmitters that diffuse across a tiny gap, a synaptic cleft, to bind with receptors on an adjacent cell, such as another neuron or a muscle cell, orchestrating thought and movement. In juxtacrine signaling, communication occurs through direct contact between the signaling and responding cells, a handshake between cells. Finally, hormones are ligands that embark on long journeys, traveling through the circulatory systems of animals or the vascular systems of plants to reach their distant target cells. Once a ligand binds with its specific receptor, it initiates a cascade of events that can profoundly influence the behavior of the target cell. For instance, neurotransmitters binding with an ionotropic receptor can rapidly alter the excitability of a target cell, generating an electrical signal. Other receptor types include protein kinase receptors, such as the receptor for the hormone insulin, and G-protein-coupled receptors, which, when activated, can trigger complex intracellular signaling cascades involving second messengers. The entire process by which a chemical or physical signal is transmitted through a cell as a series of molecular events is known as signal transduction, a marvel of biological communication. The cycle of life, at its most fundamental, is also about division. The cell cycle is a precisely choreographed series of events that occur within a cell, ultimately leading to its division into two daughter cells. These events encompass the crucial duplication of the cell's DNA and some of its organelles, followed by the meticulous partitioning of its cytoplasm into those two new cells, a process called cell division. In eukaryotes, the cells of animals, plants, fungi, and protists, there are two distinct and vital types of cell division, mitosis and meiosis. Mitosis is an integral part of the cell cycle, where replicated chromosomes are meticulously separated into two new, genetically identical nuclei. This process gives rise to daughter cells that are genetically identical to the parent cell, maintaining the same total number of chromosomes. Generally, mitosis, the division of a nucleus, is preceded by the S phase of interphase, during which the cell's DNA is replicated. It is often followed by telophase and cytokinesis, the physical division of the cytoplasm, organelles, and cell membrane into two distinct new cells, each receiving roughly equal shares of these cellular components. The various stages of mitosis collectively define the mitotic phase of a eukaryotic cell cycle, the remarkable division of a mother cell into two genetically identical daughter cells. The cell cycle is a vital process, enabling a single-celled fertilized egg to develop into a mature, complex organism, and it is also the ongoing process by which our hair, skin, blood cells, and many internal organs are constantly renewed. After cell division, each daughter cell embarks on the interphase of its own new cycle. In stark contrast to mitosis, meiosis is a specialized form of cell division that results in 4 haploid daughter cells. This is achieved through one round of DNA replication, followed by 2 distinct divisions. In the first meiotic division, meiosis 1, homologous chromosomes are separated. In the second division, meiosis 2, sister chromatids are separated. Both of these cell division cycles are essential components of sexual reproduction at some point in the life cycles of eukaryotes, and both are believed to have been present in the last eukaryotic common ancestor, underscoring their profound evolutionary significance. Prokaryotes, the archaea and bacteria, also undergo cell division, but through a simpler process called binary fission. Unlike the complex choreography of mitosis and meiosis in eukaryotes, binary fission in prokaryotes occurs without the formation of a spindle apparatus. Before binary fission, the bacterium's circular DNA is tightly coiled. It then uncoils and duplicates, with the 2 copies pulled to separate poles of the bacterium as it enlarges in preparation for splitting. The growth of a new cell wall, triggered by the polymerization of FTSE protein and the formation of a zooring, begins to pinch the bacterium in two. This new cell wall, called a septum, fully develops, resulting in the complete division of the bacterium. The 2 new daughter cells each receive a tightly coiled DNA rod, ribosomes, and plasmids, ready to embark on their own lives. Meiosis, that specialized form of cell division, is a central feature of sexual reproduction in eukaryotes. Its most fundamental function appears to be the conservation of the integrity of the genome as it is passed from parents to progeny. 2 key aspects of sexual reproduction, meiotic recombination, the shuffling of genetic material, and outcrossing, cross-fertilization, are likely maintained by powerful adaptive advantages. Recombinational repair of genomic DNA damage, where genetic material is exchanged to fix breaks, is one such advantage. Another is genetic complementation, which effectively masks the expression of deleterious recessive mutations by pairing them with healthy dominant alleles from another parent. This beneficial effect of genetic complementation, derived from outcrossing, is also known as hybrid vigor or heterosis. Charles Darwin himself, in his 1878 book, The Effects of Cross and Self-Fertilization in the Vegetable Kingdom, eloquently noted that generally cross-fertilization is beneficial and self-fertilization often injurious for the plants he studied. Ultimately, the genetic variation that is a natural byproduct of sexual reproduction, particularly through outcrossing, provides long-term advantages to lineages that engage in it, allowing them to adapt and thrive in ever-changing environments. This brings us to genetics, the profound scientific study of inheritance, the very mechanism by which life's blueprint is passed from one generation to the next. Mendelian inheritance, specifically, describes the elegant principles by which genes and traits are transmitted from parents to offspring. It operates on several key principles. The first is that genetic characteristics, or alleles, are discrete units with alternate forms. Think purple versus white flowers, or tall versus dwarf plants, each inherited from one of the two parents. The law of dominance and uniformity dictates that some alleles are dominant while others are recessive. An organism possessing at least one dominant allele will display the phenotype associated with that dominant allele. During the formation of gametes, sperm and egg cells, the alleles for each gene segregate, meaning each gamete carries only one allele for each gene. Heterozygous individuals, possessing two different alleles for a trait, produce gametes with an equal frequency of these two alleles. Finally, the law of independent assortment states that genes for different traits can segregate independently during gamete formation, implying that they are unlinked. However, there are exceptions, such as sex-linked traits, where genes located on sex chromosomes exhibit different inheritance patterns. Experimental tools like test crosses can be performed to determine the underlying genotype of an organism displaying a dominant phenotype. And the venerable Punnett square remains a simple yet powerful tool to predict the results of such crosses. The chromosome theory of inheritance, which posits that genes reside on chromosomes, received strong support from Thomas Morgan's groundbreaking experiments with fruit flies, which definitively established the sex linkage between eye color and sex in these insects. At the heart of inheritance lies the gene, a fundamental unit of heredity. A gene corresponds to a specific region of deoxyribonucleic acid, or DNA, which carries the genetic information that ultimately controls the form or function of an organism. DNA itself is a marvel of molecular architecture, composed of two polynucleotide chains that elegantly coil around each other to form the iconic double helix. In eukaryotes, DNA is organized into linear chromosomes, predominantly found within the cell nucleus. In prokaryotes, it typically exists as a circular chromosome within a region called the nucleoid. The complete set of chromosomes within a cell is collectively known as its genome, and the specific genetic makeup of an organism, its entire collection of genes, is called its genotype. DNA replication is a semi-conservative process, a clever mechanism whereby each existing strand of the double helix serves as a precise template for the synthesis of a new, complementary strand of DNA. This ensures that genetic information is faithfully copied with each cell division. However, this process isn't always perfect. Mutations are heritable changes in DNA, alterations in the genetic code. They can arise spontaneously as a result of replication errors that escape the cell's intricate proofreading and repair mechanisms, or they can be induced by environmental mutagens, such as certain chemicals, like nitrous acid or benzopyrene, or various forms of radiation, like X-rays, gamma rays, ultraviolet radiation, or particles emitted by unstable isotopes. These mutations can lead to observable phenotypic effects, ranging from loss of function, where a gene's product is no longer active, to gain of function, where a gene's product acquires a new or enhanced activity, or even conditional mutations, whose effects are only apparent under specific environmental conditions. While some mutations can be harmful, particularly if they compromise genes essential for survival, others are profoundly beneficial. Indeed, mutations are the ultimate source of genetic variation, the raw material upon which evolution acts, driving the continuous adaptation and diversification of life. The journey from the genetic blueprint to a functioning organism is orchestrated through gene expression. This is the molecular process by which the genotype, encoded in DNA, gives rise to the observable phenotype, the proteins that compose an organism's body and carry out its functions. This fundamental flow of genetic information is famously summarized by the central dogma of molecular biology, formulated by Francis Crick in 1958. Genetic information flows from DNA to RNA to protein. This process involves two main steps: transcription, where the DNA sequence is copied into an RNA molecule, and translation, where the RNA molecule serves as a template to synthesize a protein. The regulation of gene expression is exquisitely complex, influenced by both environmental factors and the various stages of development. This regulation can occur at nearly every step of the process. During transcription, determining whether a gene is copied into RNA. During RNA splicing, where non-coding regions are removed from the RNA. During translation, controlling the rate at which RNA is converted into protein. And even through post-translational modification, where proteins are altered after they are synthesized. Gene expression can be influenced by both positive and negative regulation. In positive regulation, activator proteins, a type of transcription factor, bind to DNA sequences near or at a gene's promoter, stimulating transcription. In negative regulation, repressor proteins, another type of transcription factor, bind to a DNA sequence called an operator, which is part of an operon, to block transcription. Repressors can sometimes be inhibited by compounds called inducers, like allolactose, which then allows transcription to proceed. Genes that can be activated by inducers are called inducible genes, in contrast to constitutive genes that are almost constantly active. Structural genes, on the other hand, encode proteins that are not involved in gene regulation themselves. Beyond these regulatory events involving promoters and operators, gene expression can also be finely tuned by epigenetic changes to chromatin, the complex of DNA and protein found in eukaryotic cells, which can influence how accessible genes are for transcription without altering the underlying DNA sequence. The interplay between genes, development, and evolution is a profound narrative in biology. Development is the intricate process by which a multicellular organism, be it a plant or an animal, undergoes a series of remarkable changes, beginning from a single cell and progressively taking on the diverse forms characteristic of its life cycle. Four key processes underpin this transformation: determination, differentiation, morphogenesis, and growth. Determination sets the developmental fate of a cell, a fate that becomes progressively more restrictive as development proceeds. Differentiation is the process by which specialized cells, such as nerve cells, muscle cells, or skin cells, arise from less specialized cells, like stem cells. Stem cells are remarkable, undifferentiated or partially differentiated cells that possess the extraordinary ability to differentiate into various cell types and to proliferate indefinitely, producing more of the same stem cell. Cellular differentiation dramatically alters a cell's size, shape, membrane potential, metabolic activity, and responsiveness to signals. These changes are largely due to highly controlled modifications in gene expression and epigenetics, rather than changes in the DNA sequence itself, with only a few rare exceptions. Thus, different cells within the same organism can have vastly different physical characteristics despite possessing the exact same genome. Morphogenesis, the development of body form, is a breathtaking result of spatial differences in gene expression. A small fraction of the genes in an organism's genome forms what is known as the developmental genetic toolkit. These toolkit genes are the master controllers of an organism's development. What is truly astonishing is how highly conserved these toolkit genes are among different phyla, meaning they are ancient and remarkably similar in widely separated groups of animals, underscoring common ancestry. Differences in how these toolkit genes are deployed, when, where, and how much they are expressed profoundly affect the body plan, as well as the number, identity, and pattern of body parts. Among the most important of these toolkit genes are the Hox genes, which determine where repeating structures, such as the numerous vertebrae of snakes, will grow in a developing embryo or larva. These genes are the silent directors of anatomical diversity. Ultimately, evolution is the central organizing concept in all of biology. It is the undeniable change in heritable characteristics of populations over successive generations, the ongoing saga of life's transformation. The concept of artificial selection, where humans selectively breed animals or plants for specific traits, provided Darwin with a powerful analogy. He recognized that if humans could exert such influence, then in the natural world, nature itself played the role of the selector. Given that traits are inherited, populations inherently contain a varied mix of these traits, and that reproduction has the potential to increase any population beyond available resources, Darwin powerfully argued that individuals possessing heritable traits that better adapted them to their environments were more likely to survive and produce more offspring than others. This, he inferred, would lead to the gradual accumulation of favorable traits over successive generations, thereby continuously increasing the match between organisms and their environment, a beautiful and powerful process we call natural selection. This process of change eventually leads to speciation, the remarkable process by which one evolutionary lineage splits into two distinct lineages, evolving independently from each other. For speciation to occur, a critical prerequisite is reproductive isolation, meaning the two groups can no longer successfully interbreed. Reproductive isolation can arise from various incompatibilities between genes, as described by the Bateson-Dobzhansky Muller model, where the accumulation of genetic differences leads to hybrid inviability or sterility. Indeed, reproductive isolation tends to increase with increasing genetic divergence. One common mechanism of speciation occurs when physical barriers, such as mountain ranges or oceans, divide an ancestral species, isolating populations from each other. This is known as allopatric speciation, the formation of new species in different homelands. To fully grasp the grand narrative of evolution, we turn to phylogeny, the evolutionary history of a specific group of organisms or their genes. This history can be beautifully represented using a phylogenetic tree, a diagram illustrating the lines of descent among organisms or their genes. Each line drawn on the time axis of a tree represents a lineage, tracing the descendants of a particular species or population. When a lineage divides into two, it is depicted as a fork or split on the phylogenetic tree, representing a speciation event. Phylogenetic trees form the fundamental basis for comparing and grouping different species. Different species that share a feature inherited from a common ancestor are said to possess homologous features, or synapomorphies, indicating their shared evolutionary heritage. This phylogenetic framework provides the very basis of biological classification. This classification system is hierarchical and rank-based, with the highest rank being the domain, followed by kingdom, phylum, class, order, family, genus, and finally, species. All known organisms on Earth can be classified into one of three overarching domains: archaea, bacteria, and eukarya, which includes the familiar kingdoms of fungi, plants, and animals. The history of life on Earth is an epic saga, tracing the journey of organisms from the earliest emergence of life to the astonishing diversity we witness today. Our planet formed approximately 4.5 billion years ago, and all life on Earth, both living and extinct, is believed to have descended from a single last universal common ancestor that lived roughly 3.5 billion years ago. Geologists have meticulously developed a geologic time scale, dividing Earth's vast history into major divisions, beginning with four eons: the Hadean, Archean, Proterozoic, and Phanerozoic. The first three are collectively known as the Precambrian, a colossal span lasting approximately 4 billion years. Each eon is further subdivided into eras, with the Phanerozoic eon, which began 539 million years ago, being partitioned into the Paleozoic, Mesozoic, and Cenozoic eras. These three eras, in turn, encompass 11 geological periods, from the Cambrian through to the Quaternary. The remarkable similarities observed among all known present-day species stand as compelling evidence that they have diverged through the process of evolution from this common ancestor. Biologists regard the ubiquity of the genetic code itself, its near universality across bacteria, archaea, and eukaryotes, as one of the most powerful pieces of evidence for universal common descent. In the early Archean Eon, microbial mats of coexisting bacteria and archaea were the dominant form of life, and it is thought that many of the major steps in early evolution unfolded within these ancient, resilient communities. The earliest robust evidence of eukaryotes dates back approximately 1.85 billion years ago, and while they may have been present earlier, their diversification dramatically accelerated once they began harnessing oxygen in their metabolism. Later, around 1.7 billion years ago, the first multicellular organisms began to appear, exhibiting differentiated cells performing specialized functions, a monumental leap in biological complexity. The earliest algae-like multicellular land plants are dated to about 1 billion years ago, although evidence suggests that microorganisms formed the very first terrestrial ecosystems at least 2.7 billion years ago, paving the way for the eventual colonization of land by plants during the Ordovician period. These early land plants were so incredibly successful that their photosynthetic activity is thought to have contributed significantly to the late Devonian extinction event by dramatically altering atmospheric composition. The enigmatic Ediacara biota appeared during the Ediacaran period, followed by an explosion of life during the Cambrian period, approximately 525 million years ago, where vertebrates and most other modern animal phyla originated in what is known as the Cambrian explosion. During the Permian period, synapsids, which include the ancestors of mammals, dominated the terrestrial landscape. However, most of this diverse group perished in the catastrophic Permian Triassic extinction event 252 million years ago, one of the most severe mass extinctions in Earth's history. During the subsequent recovery from this cataclysm, archosaurs rose to prominence, becoming the most abundant land vertebrates. One group of archosaurs, the dinosaurs, reigned supreme throughout the Jurassic and Cretaceous periods. After the devastating Cretaceous Paleogene extinction event 66 million years ago, which extinguished the non-avian dinosaurs, mammals underwent a rapid increase in size and diversity, seizing the ecological opportunities left vacant. Such mass extinctions, while destructive, paradoxically may have accelerated evolution by creating ecological vacuums that allowed new groups of organisms to diversify rapidly. The diversity of life on Earth is truly staggering, a testament to billions of years of evolution. Let's delve into the three great domains of life. Bacteria constitute a vast and ancient domain of prokaryotic microorganisms. Typically just a few micrometers in length, bacteria exhibit a remarkable array of shapes, from spherical to rod-like and spiral. They were among the very first life forms to appear on Earth and are now ubiquitous, present in virtually every habitat imaginable: soil, water, acidic hot springs, radioactive waste, and even the deep biosphere of Earth's crust. Bacteria also engage in intricate symbiotic and parasitic relationships with plants and animals, including ourselves. Despite their omnipresence, most bacteria remain uncharacterized, with only about 27% of bacterial phyla having species that can be cultured in a laboratory, hinting at a vast, unexplored microbial world. Archaea represent the other domain of prokaryotic cells. Initially, they were classified as bacteria, even given the name archaebacteria, but this term has largely fallen out of use as we've recognized their unique nature. Archaeal cells possess distinct properties that clearly separate them from both bacteria and eukaryotes. While generally similar to bacteria in size and shape, though some archaea, like the flat, square cells of Haloquadratum walsbeii, exhibit truly bizarre forms. Archaea possess genes and several metabolic pathways that are more closely related to those of eukaryotes, particularly for enzymes involved in transcription and translation. Other aspects of archaeal biochemistry are entirely unique, such as their reliance on ether lipids in their cell membranes, including molecules like archaeols. Archaea also display an incredible metabolic versatility, utilizing a wider range of energy sources than eukaryotes, from organic compounds like sugars to ammonia, metal ions, or even hydrogen gas. Salt-tolerant archaea, known as halorchaea, can even use sunlight as an energy source, and some species of archaea fix carbon. However, unlike plants and cyanobacteria, no known species of archaea performs both photosynthesis and carbon fixation. Archaea reproduce asexually through binary fission, fragmentation, or budding, and notably, unlike bacteria, no known species of archaea forms endospores. The first observed archaea were extremophiles, creatures thriving in extreme environments like hot springs and salt lakes, where no other organisms could survive. However, the development of improved molecular detection tools has revealed archaea in almost every habitat on Earth, including soil, oceans, and marshlands. They are particularly numerous in the oceans, where archaea in plankton may be one of the most abundant groups of organisms on the planet. Archaea are a major, often overlooked component of Earth's life. They are integral to the microbiota of all organisms, playing crucial roles in the human gut, mouth, and on our skin. Their immense morphological, metabolic, and geographical diversity allows them to fulfill a multitude of ecological roles: fixing carbon, cycling nitrogen, turning over organic compounds, and maintaining complex microbial symbiotic and syntrophic communities, highlighting their fundamental importance to global biogeochemical cycles. Then we come to the eukaryotes, a domain hypothesized to have emerged from an archaeal ancestor, followed by crucial endosymbioses with bacteria. These ancient symbiotic events gave rise to mitochondria and chloroplasts, both of which are now integral parts of modern eukaryotic cells. The major lineages of eukaryotes diversified during the Precambrian, approximately 1.5 billion years ago, and can be broadly classified into 8 major clades: alveolates, excavates, straminopiles, plants, rhizarians, amoebozoants, fungi, and animals. 5 of these clades are collectively known as protists, a somewhat artificial grouping of mostly microscopic eukaryotic organisms that are not plants, fungi, or animals. While it's highly probable that protists share a common ancestor, the protist grouping itself is not a formal taxonomic clade, as some protists are more closely related to plants, fungi, or animals than they are to other protists. Like terms such as algae or invertebrates, protist is primarily a term of convenience, with most being unicellular, referred to as microbial eukaryotes. Plants, those vital producers, are primarily multicellular organisms, predominantly photosynthetic eukaryotes belonging to the kingdom Plantae, a definition that excludes fungi and some algae. Plant cells themselves originated from an incredible endosymbiosis event about 1 billion years ago, when an early eukaryote engulfed a cyanobacterium, which then evolved into the chloroplast. The first clades to emerge after this primary endosymbiosis were aquatic, and most aquatic photosynthetic eukaryotic organisms are broadly described as algae. Like protists, algae is a term of convenience, as not all algal groups are closely related. Algae encompass several distinct clades, such as glaucophytes, which are microscopic freshwater algae that may resemble the early unicellular ancestor of Plantae. Unlike glaucophytes, other algal clades, such as red and green algae, are typically multicellular. Green algae comprise 3 major clades: chlorophytes, coleochaetophytes, and stoneworts, each contributing to the rich diversity of aquatic photosynthesis. Fungi, another fascinating kingdom, are eukaryotes that uniquely digest food outside their bodies. They secrete digestive enzymes that break down large food molecules externally before absorbing the resulting nutrients through their cell membranes. Many fungi are also saprobes, meaning they feed on dead organic matter, making them crucial decomposers in virtually all ecological systems, tirelessly recycling nutrients. Animals, the kingdom we belong to, are multicellular eukaryotes. With very few exceptions, animals consume organic material, breathe oxygen, possess the remarkable ability to move, typically reproduce sexually, and develop from a hollow sphere of cells called a blastula during their embryonic development. Over 1.5 million living animal species have been formally described, with an astonishing approximately 1 million being insects. But estimates suggest there are likely over 7 million animal species in total, a vast and largely unexplored realm of life. Animals engage in incredibly complex interactions with each other and their environments, forming the intricate food webs that define ecosystems. Finally, we confront the enigmatic viruses. Viruses are submicroscopic infectious agents that can only replicate inside the living cells of other organisms. They infect all known types of life forms, from animals and plants to microorganisms, including bacteria and archaea. More than 6,000 virus species have been described in detail, but this is undoubtedly a tiny fraction of the true viral diversity. Viruses are found in almost every ecosystem on Earth and are, in fact, the most numerous type of biological entity on the planet. The origins of viruses and the evolutionary history of life remain a subject of active research and debate. Some may have evolved from plasmids, small pieces of DNA that can move between cells, while others may have evolved from more complex cellular ancestors. In the grand scheme of evolution, viruses are important drivers of horizontal gene transfer, a process that significantly increases genetic diversity in a way analogous to, yet distinct from, sexual reproduction. Because viruses possess some, but not all, of the universally accepted characteristics of life, they have been compellingly described as organisms at the edge of life, or simply as fascinating self-replicators that blur the lines between living and non-living. All of this life, in its boundless forms and functions, exists within a dynamic framework known as ecology, the scientific study of the distribution and abundance of life, and the intricate interactions between organisms and their environment. The community of living or biotic organisms, in conjunction with the non-living or abiotic components of their environment, factors like water, light, radiation, temperature, humidity, atmosphere, acidity, and soil, form what we call an ecosystem. These biotic and abiotic components are inextricably linked through nutrient cycles and energy flows. Energy, primarily from the sun, enters the system through the miraculous process of photosynthesis and is incorporated into plant tissue. Animals, by feeding on plants and on each other, then move this matter and energy through the system, creating intricate food webs. They also profoundly influence the quantity of plant and microbial biomass present. Decomposers, those essential recyclers, break down dead organic matter, releasing carbon back to the atmosphere and facilitating nutrient cycling by converting nutrients stored in dead biomass back into forms that can be readily used by plants and other microbes, completing the grand cycle of life. Within ecosystems, we study populations, groups of organisms of the same species that occupy a specific area and reproduce from generation to generation. The size of a population can be estimated by multiplying its density by the area or volume it inhabits. A crucial ecological concept is the carrying capacity of an environment, which represents the maximum population size of a species that can be sustained indefinitely by that specific environment, given the available food, habitat, water, and other vital resources. The carrying capacity of a population is not static. It can be significantly affected by changing environmental conditions, such as shifts in resource availability and the metabolic cost of maintaining those resources. In human populations, technological advancements, such as the Green Revolution, have historically helped to increase Earth's carrying capacity for humans, defying past predictions of impending population decline, such as those famously made by Thomas Malthus in the 18th century. Populations do not exist in isolation. They form communities, groups of populations of different species occupying the same geographical area at the same time. Within these communities, biological interactions, the effects that pairs of organisms living together have on each other, are constant. These interactions can be intraspecific, occurring between individuals of the same species, or interspecific, occurring between individuals of different species. These effects can be short-term, like pollination or predation, or long-term, forming symbiotic relationships. Both short-term and long-term interactions often strongly influence the evolution of the species involved. A long-term interaction is called a symbiosis, and these range from mutualism, where both partners benefit, to competition, where both partners are harmed. Every species participates as either a consumer, a resource, or both in consumer-resource interactions, which form the intricate core of food chains and food webs. Within any food web, there are distinct trophic levels. At the lowest level are the primary producers, or autotrophs, such as plants and algae, which convert energy and inorganic material into organic compounds. These organic compounds then become the energy source for the rest of the community. At the next level are the heterotrophs, species that obtain energy by breaking apart organic compounds from other organisms. Heterotrophs that consume plants are called primary consumers, or herbivores. Heterotrophs that consume herbivores are secondary consumers, or carnivores. And those that eat secondary consumers are tertiary consumers, and so on. Omnivorous heterotrophs are versatile, able to consume at multiple trophic levels. Finally, the crucial role of decomposers is to feed on the waste products or dead bodies of organisms, recycling nutrients back into the ecosystem. On average, the total amount of energy incorporated into the biomass of a trophic level per unit of time is only about 1/10 of the energy from the trophic level it consumes. The remaining 90% of energy is lost as waste and dead material, which feeds decomposers, and as heat lost from metabolic processes, highlighting the inherent inefficiency of energy transfer through food webs. Zooming out to the largest scale, we consider the global ecosystem, or biosphere, where matter exists in different interacting compartments. These compartments can be biotic or abiotic, and accessible or inaccessible, depending on their forms and locations. For example, matter within terrestrial autotrophs is both biotic and accessible to other organisms, while matter locked within rocks and minerals is abiotic and largely inaccessible. A biogeochemical cycle describes the pathway by which specific elements of matter are turned over or moved through the biotic compartments, the biosphere, and the abiotic compartments, the lithosphere, atmosphere, and hydrosphere, of Earth. There are vital biogeochemical cycles for essential elements such as nitrogen, carbon, and water, each a complex, interconnected dance that sustains our living planet. In light of this incredible diversity and interconnectedness, conservation biology has emerged as a critical discipline. It is the scientific study of the conservation of Earth's biodiversity, driven by the urgent aim of protecting species, their habitats, and entire ecosystems from excessive rates of extinction and the erosion of crucial biotic interactions. It investigates the factors that influence the maintenance, loss, and restoration of biodiversity, and seeks to understand the science of sustaining the evolutionary processes that generate genetic, population, species, and ecosystem diversity. The profound concern stems from alarming estimates suggesting that up to 50% of all species on the planet could disappear within the next 50 years, a catastrophic loss with far-reaching consequences, contributing to poverty, starvation, and fundamentally altering the future course of evolution on this planet. Biodiversity is not merely an aesthetic luxury. It profoundly affects the functioning of ecosystems, which in turn provide a multitude of vital services upon which all human societies depend. Conservation biologists tirelessly research and educate on the alarming trends of biodiversity loss, species extinctions, and the devastating negative effects these are having on our collective ability to sustain the well-being of human society. In response to this looming biodiversity crisis, organizations and concerned citizens worldwide are actively engaged in conservation action plans, directing essential research, monitoring efforts, and educational programs that address these critical concerns at scales ranging from local communities to global initiatives. The future of life, in all its astonishing forms, hinges on our understanding and action. Welcome back, fellow travelers through the cosmos and the microscopic realms. Settle in, find your comfortable space, and let your mind unfurl as we continue our journey into the very essence of life itself. Last time, we laid the groundwork, marveling at how the universe's fundamental building blocks, from scattered cosmic dust to the intricate architecture of living cells, began to coalesce. Tonight, we delve deeper into the bustling, energetic heart of every single living thing, the engine of life, unraveling cellular metabolism and energy flow. Imagine, if you will, a city that never sleeps. Not just any city, but one teeming with billions of tiny inhabitants, each a miniature factory operating with breathtaking precision. This city is the single cell, and the constant hum of activity within its walls is what we call metabolism. It's the grand symphony of chemical reactions, orchestrated by countless enzymes, that allows life to not merely exist, but to thrive, to grow, to adapt, and to endlessly perpetuate itself. From the smallest bacterium to the largest blue whale, every organism is engaged in this ceaseless metabolic dance. It is the fundamental process that converts raw materials into the energy needed to power every single cellular function. Think of it as the cell's personal power grid, its intricate waste management system, and its dedicated construction crew, all rolled into one impossibly small, self-regulating package. At its core, metabolism serves three critical purposes. First, to transform the fuel we consume, whether it's sunlight for a plant or a meal for an animal, into usable energy for the cell's endless demands. Second, it breaks down these fuels into the monomer building blocks, the molecular Lego bricks, that cells use to construct everything from new proteins to vital genetic material. And third, it efficiently eliminates the metabolic wastes, the byproducts of all this tireless activity, ensuring the cellular environment remains pristine and functional. These reactions, thousands upon thousands of them, don't happen chaotically. They're meticulously organized into what we call metabolic pathways, like a series of interconnected assembly lines. In each step of these pathways, one chemical compound is transformed into another, and for every single transformation, there's a dedicated specialist, a specific enzyme. These enzymes are the unsung heroes of life, acting as catalysts. They don't get used up in the reaction, but they dramatically speed up the process, enabling reactions that would otherwise take eons to occur in milliseconds. Without them, life as we know it simply wouldn't be possible. They are the precision tools that allow organisms to conduct these energy-demanding reactions by cleverly coupling them with other spontaneous reactions that release energy. It's an exquisite balance of energy capture and expenditure. Metabolism is generally divided into two broad categories, like two sides of the same biological coin: catabolism and anabolism. Catabolism is the grand process of breaking down. It's the deconstruction crew, taking large, complex molecules like the glucose you might find in a sugar molecule and systematically dismantling them into smaller, simpler components. This breakdown releases energy, much like carefully dismantling a finely tuned machine might allow you to reclaim its component parts and the kinetic energy involved in its operation. Anabolism, on the other hand, is the building up, the construction phase. It's where the cell takes those smaller, simpler building blocks liberated by catabolism and synthesizes new, complex compounds, proteins, carbohydrates, lipids, nucleic acids, everything the cell needs to grow, repair, and reproduce. This process, as you might imagine, consumes energy, much like building something requires an input of effort and resources. The elegance lies in the interconnectedness, The energy released by catabolic reactions fuels the anabolic ones. It's a closed-loop system, remarkably efficient and endlessly self-sustaining. Now, let's talk about the beating heart of this energy transformation, cellular respiration. If metabolism is the entire intricate city, cellular respiration is its bustling power plant. It's the set of metabolic reactions that convert the chemical energy stored in nutrients, like that glucose we mentioned, into the universal energy currency of the cell, adenosine triphosphate, or ADP. Think of ADP as the fully charged battery that powers every cellular activity, from muscle contraction to the intricate synthesis of new molecules. The reactions involved in respiration are primarily catabolic, systematically breaking down larger molecules to release their stored energy. While technically a combustion reaction, the controlled burning of fuel, it's a far cry from the explosive release of energy you'd see in an actual fire. Inside a cell, this energy release is slow, controlled, and exquisitely managed, preventing damage and maximizing efficiency. For animal and plant cells, glucose is the primary fuel. And when oxygen is available, we enter the sophisticated world of aerobic respiration, a four-stage masterpiece of biochemical engineering. It all begins in the cell's cytoplasm with glycolysis, a term that literally means sugar splitting. Here, a single molecule of glucose is meticulously broken down into two molecules of pyruvate. This initial skirmish yields a modest, but crucial, two net molecules of ADP. From the cytoplasm, each pyruvate molecule then makes its way into the mitochondria, the cell's true powerhouses, where it's oxidized into acetyl-CoA, releasing carbon dioxide and generating another energy-carrying molecule called NADH. Next up, the citric acid cycle, also known as the Krebs cycle. This intricate cycle unfolds within the mitochondrial matrix, a series of chemical reactions that further dismantle the acetyl-CoA. This stage doesn't produce a massive amount of ADP directly, but it's a critical preparatory step, generating a rich harvest of those electron-carrying molecules, NADH and FADH2. These molecules are like tiny, highly charged shuttles, brimming with potential energy, ready for the final, most productive stages of respiration. And that brings us to the grand finale, the most prolific ADP-generating stage, oxidative phosphorylation, which occurs in the inner membrane folds of the mitochondria, known as the cristae. This stage is a two-part marvel, beginning with the electron transport chain. Imagine a series of four protein complexes, like a biochemical staircase, embedded within the mitochondrial membrane. The NADH and FADH2 molecules arrive, drop off their electrons, and these electrons are then passed from one protein complex to the next, much like a bucket brigade. With each transfer, a small amount of energy is released. This released energy is not wasted. It's cleverly harnessed to pump protons, hydrogen ions, from the mitochondrial matrix across the inner membrane into the intermembrane space. This creates a powerful concentration gradient, a veritable dam of protons, known as the proton motive force. And here's where the true genius lies. As these protons, driven by their concentration gradient, rush back across the membrane through a specialized enzyme called ADP synthase, their flow powers this molecular turbine, synthesizing a huge number of ADP molecules by phosphorylating ADP. It's a breathtakingly efficient process, almost like a miniature hydroelectric power plant within your cells. The journey of these electrons eventually culminates with molecular oxygen acting as the final electron acceptor, forming water as a harmless byproduct. But what if oxygen isn't available? What happens then? When oxygen is scarce, cells switch gears, engaging in a different metabolic strategy, fermentation. In these anaerobic conditions, pyruvate doesn't enter the mitochondria. Instead, it remains in the cytoplasm. Here, it's converted into various waste products, which are then expelled from the cell. The primary purpose of fermentation is not to generate vast quantities of ADP. It produces very little, but rather to regenerate those crucial electron carriers, specifically NAD+, so that glycolysis can continue. Without NAD+, glycolysis would grind to a halt, and even the small amount of ADP it produces would cease, leading to cellular energy starvation. The specific waste product generated during fermentation depends on the organism. In our own skeletal muscles, during strenuous exercise when oxygen demand outstrips supply, pyruvate is converted into lactic acid. This is what contributes to that burning sensation you feel during an intense workout. Once oxygen becomes available again during recovery, the lactic acid can be converted back to pyruvate and enter aerobic respiration. In yeast, the waste products are ethanol and carbon dioxide, the very process that gives us bread and alcoholic beverages. So, even in the absence of oxygen, life finds a way to extract energy, albeit less efficiently. Now, let's pivot from consuming energy to creating it. For life to truly flourish, there needs to be a fundamental source of energy, and for most of Earth's ecosystems, that source is the sun. This brings us to photosynthesis, the miraculous process used by plants, algae, and cyanobacteria to convert light energy into chemical energy. This chemical energy is then stored in carbohydrate molecules, like sugars, synthesized from carbon dioxide and water, often releasing oxygen as a waste product. Photosynthesis is the ultimate life-sustaining process on our planet, largely responsible for the oxygen-rich atmosphere we breathe and the vast majority of the energy that fuels all life. Photosynthesis, much like cellular respiration, is a multi-stage marvel. It begins with light absorption. Chlorophyll pigments, the molecules that give plants their green hue, absorb light energy within specialized structures called thylakoid membranes inside chloroplasts. This absorbed light energy is then used to extract electrons from a donor molecule, in most cases, water. This splitting of water not only liberates electrons, but also releases oxygen, the vital gas we breathe. These excited electrons then embark on an electron transport journey, similar in principle to what we saw in respiration, but operating in reverse. They move through a series of electron carriers, eventually reaching a final electron acceptor, reducing NADP+ into NADPH, another crucial energy-carrying molecule. This electron transport is intricately linked to the pumping of protons, creating a pH gradient across the thylakoid membrane, much like the proton motive force in mitochondria. And just like in respiration, this proton gradient drives ADP synthesis. Protons flow down their concentration gradient through an ADP synthase, synthesizing ADP. The NADPH and ADP generated during these light-dependent reactions are then ready for the final stage, carbon fixation, also known as the Calvin cycle. This stage takes place independently of light, hence light-independent or dark reactions, and it's where the magic of sugar creation truly happens. Atmospheric carbon dioxide is fixed into existing organic carbon compounds, ultimately leading to the synthesis of glucose. This is where the energy from sunlight is finally locked away in the chemical bonds of sugar, ready to be utilized by the plant or consumed by other organisms. Beyond these grand energy transformations, the cell is a master communicator. Cell signaling, or cell communication, is the ability of cells to receive, process, and transmit signals, both from their external environment and from within themselves. These signals can be diverse: light, electrical impulses, heat, but most commonly, they are chemical signals or ligands that interact with specific receptors. These receptors can be embedded in the cell membrane, or they can be located deep inside the cell. There are various sophisticated ways cells communicate. In autocrine signaling, a cell releases a ligand that acts back upon itself. Think of a tumor cell, which can unfortunately release signals that initiate its own uncontrolled division. Paracrine signaling involves ligands diffusing to nearby cells, influencing their behavior. Neurons, for instance, release neurotransmitters that diffuse across a tiny gap called a synaptic cleft to bind with receptors on an adjacent neuron or muscle cell. Juxtacrine signaling is the most direct, requiring actual physical contact between the signaling and responding cells. And finally, hormones are ligands that travel through the circulatory systems of animals or the vascular systems of plants, reaching distant target cells to exert their influence. Once a ligand binds to its receptor, it triggers a cascade of molecular events within the cell, a process known as signal transduction. This can lead to a vast array of changes in cellular behavior, from altering excitability to initiating complex gene expression patterns. It's how cells coordinate their activities, respond to threats, and maintain the delicate balance of life. And speaking of balance and coordination, we arrive at the cell cycle, the series of events that culminates in a cell dividing into two daughter cells. This isn't a simple split. It's a meticulously choreographed process involving the duplication of the cell's DNA and organelles, followed by the precise partitioning of its contents. In eukaryotic cells, the cells of animals, plants, fungi, and protists, there are two distinct types of cell division: mitosis and meiosis. Mitosis is the process by which a single cell divides into two genetically identical daughter cells. This is how organisms grow, repair damaged tissues, and replace old cells. It's a vital, ongoing process in multicellular organisms, ensuring that every new hair, skin cell, or blood cell carries the exact same genetic blueprint. The mitotic phase is just one part of the broader cell cycle, which includes a preparatory period called interphase, during which the cell grows and, crucially, duplicates its DNA. Mitosis itself involves the replicated chromosomes being precisely separated into two new nuclei, followed by cytokinesis, the division of the cytoplasm and organelles, yielding two complete, functional daughter cells. Meiosis, in contrast to mitosis, is a specialized form of cell division that results in 4 haploid daughter cells, cells with half the number of chromosomes of the parent cell. This reduction in chromosome number is absolutely essential for sexual reproduction. It involves one round of DNA replication followed by 2 divisions. In the first meiotic division, homologous chromosomes are separated, and in the second, sister chromatids are separated. Both types of cell division are integral to the incredible diversity and perpetuation of life. Meiosis, with its accompanying genetic recombination and outcrossing, is believed to be a fundamental mechanism for maintaining genomic integrity and providing the genetic variation upon which evolution can act. As Charles Darwin himself observed, cross-fertilization is often beneficial, providing the robust genetic mixing that fuels adaptation and resilience. Even prokaryotes, the single-cell bacteria and archaea, undergo cell division, though it's a simpler process called binary fission. Without the complex spindle apparatus of eukaryotes, their tightly coiled DNA uncoils, duplicates, and then is pulled to opposite poles as the cell elongates. A new cell wall forms, pinching the bacterium in two, resulting in 2 genetically identical daughter cells, each a complete, self-sufficient entity. From the grand metabolic dance of catabolism and anabolism, the powerful efficiency of cellular respiration and photosynthesis, to the intricate communication networks and the precise ballet of cell division, the single cell is a universe unto itself. It is the fundamental engine of life, operating with an almost unbelievable complexity and elegance, a testament to billions of years of refinement and adaptation. And as we've seen, every one of these processes, every single enzyme, and every chemical reaction is designed to ensure the most precious outcome, the continuation of life. As you drift now, ponder the silent, ceaseless energy transformations happening within every cell of your own being. It is a profound, intricate miracle happening moment by moment, ensuring your existence and connecting you to the very first spark of life on this planet. Rest easy and perhaps dream of the incredible, invisible engines that power us all. We've journeyed through the fundamental architecture of life, from the cosmic dust that cradled its inception to the intricate cellular machinery that hums with metabolic energy. Now, as our gaze shifts, we delve into the very blueprint of existence, the genetic symphony. Imagine, if you will, an ancient scroll inscribed with the precise instructions for every living thing, from the simplest bacterium to the most complex redwood, from the ephemeral glow of a firefly to the profound intelligence of the human mind. This scroll, continuously copied and passed down through countless generations, is what we call DNA. It's the grandmaster score, conducting the entire symphony of life. Our understanding of this intricate score, this profound language of life, begins with genetics, the scientific study of inheritance. For millennia, humanity has observed the undeniable truth that offspring tend to resemble their parents, that traits are passed down through the generations. But the precise mechanisms remained veiled in mystery until a monk in a garden began to meticulously count peas. That monk was Gregor Mendel, and his work in 1865 laid the foundation for modern genetics. He wasn't working with microscopes or complex molecular tools, but with careful observation and a keen statistical mind. Mendel identified several fundamental principles of Mendelian inheritance. The idea that genetic characteristics, or alleles, are discrete units with alternate forms. Think of the purple versus white flowers, or tall versus dwarf pea plants. He posited that each individual inherits one allele from each parent. His law of dominance and uniformity explained why some traits seem to disappear in one generation only to reappear in the next, concluding that some alleles are dominant, masking the presence of recessive ones. A plant with just one dominant allele for purple flowers would indeed display purple flowers. Furthermore, Mendel discovered that during the formation of reproductive cells, or gametes, these alleles for each gene segregate, meaning each gamete receives only one allele for any given gene. And then his most profound insight, the law of independent assortment, which states that genes for different traits can segregate independently during gamete formation, implying they are unlinked. This explained the vast combinations of traits we see in offspring, the wonderful variety that arises even within a single family. While Mendel's work was revolutionary, its significance wasn't fully appreciated until the early 20th century. It was then that the groundbreaking discoveries of Darwinian evolution, with its focus on natural selection, were reconciled with Mendel's principles, forming what we now call the modern synthesis of evolutionary biology. This was a pivotal moment, weaving together two profound threads of biological understanding into a single, cohesive narrative. But the physical nature of Mendel's factors, what we now call genes, remain elusive. The next great leap came in the 1940s and early 1950s, with a series of elegant experiments, most famously by Alfred Hershey and Martha Chase. Their work pointed unmistakably to DNA as the component of chromosomes that carried these trait-carrying units. This set the stage for one of the most iconic discoveries in science. In 1953, James Watson and Francis Crick, building on the work of Rosalind Franklin and Maurice Wilkins, unveiled the magnificent double helical structure of DNA. This wasn't just a pretty picture. It was the key to understanding how genetic information is stored, copied, and passed down. The double helix, with its elegant ladder-like structure, instantly suggested a mechanism for replication, for how life could faithfully duplicate its instructions. This discovery marked the true transition into the era of molecular genetics, an era that would transform our understanding of life itself. A gene, then, is a specific region of this magnificent DNA molecule. It's a unit of heredity that carries the genetic information, the instructions that control the form or function of an organism. DNA itself is composed of two long polynucleotide chains that coil around each other, forming that iconic double helix. In eukaryotic cells, like our own, DNA is organized into linear structures called chromosomes within the cell nucleus. In simpler prokaryotic organisms, like bacteria, it often forms a circular chromosome within a region called the nucleoid. The complete set of chromosomes in a cell, all the genetic instructions, is known as its genome. The faithful copying of this genetic information is paramount, and it occurs through a process called DNA replication. This is a semi-conservative process, meaning that each strand of the original DNA molecule serves as a template for a new strand. It's like having a master copy, and each time you make a duplicate, you keep half of the original, ensuring accuracy. However, replication isn't always perfect. Sometimes, errors occur, leading to mutations. These are heritable changes in the DNA sequence. Mutations can arise spontaneously from replication errors that weren't corrected by the cell's sophisticated proofreading mechanisms, or they could be induced by environmental factors, chemicals like nitrous acid, or various forms of radiation, such as X-rays or ultraviolet light. These changes can have a wide range of effects, from silent alterations that have no impact to profound changes that lead to loss of function or even gain-of-function phenotypes. The very idea of mutations might sound concerning, but it's crucial to remember that some mutations are not only harmless, but are actually beneficial. They are the ultimate source of genetic variation, the raw material upon which evolution acts. Without mutations, without these random changes in the genetic code, life would be static, unable to adapt, unable to diversify and evolve. While harmful mutations can indeed lead to serious consequences, often through the loss of function of vital genes, the balance of beneficial and neutral mutations fuels the ongoing dance of life on Earth. So, we have the blueprint, the DNA, but how does this blueprint translate into the observable traits, the structures, and functions of an organism? This is the process of gene expression, the molecular cascade by which the genotype encoded in DNA gives rise to the observable phenotype in the proteins of an organism's body. This core principle is elegantly summarized by the central dogma of molecular biology, formulated by Francis Crick in 1958. It states that genetic information flows from DNA to RNA to protein. It's a one-way street of information transfer, a fundamental principle governing all known life. This flow involves two primary processes. First, transcription, where the DNA sequence of a gene is copied into a messenger RNA molecule. Think of it as a working copy of a specific instruction. Then, translation, where this messenger RNA is read by cellular machinery, and its sequence of information is used to assemble a chain of amino acids, forming a protein. Proteins, as we discussed earlier, are the workhorses of the cell, carrying out a vast array of functions from structural support to enzymatic catalysis. But the symphony of gene expression isn't a continuous, uncontrolled outpouring of all instructions all the time. It is a tightly controlled, exquisitely orchestrated process, finely tuned to the needs of the cell and the organism. This is where gene regulation comes into play. The expression of genes can be influenced by a myriad of factors, both environmental and developmental, at almost every step of the process. From the initial transcription of DNA into RNA, through RNA splicing, translation, and even post-translational modifications of the protein itself. Gene expression can be subject to positive or negative regulation. In positive regulation, specific regulatory proteins called activators bind to DNA sequences near a gene's promoter, stimulating transcription. It's like a conductor signaling an instrument to begin playing. Conversely, negative regulation involves repressors, another type of transcription factor, which bind to a DNA sequence called an operator, often part of a cluster of genes called an operon, particularly common in prokaryotes. When a repressor binds, it physically blocks transcription, silencing the gene, like a conductor quieting a section of the orchestra. Remarkably, these repressors can sometimes be inhibited by compounds called inducers, allowing transcription to proceed. This is how some genes are inducible, turning on only when a specific signal or nutrient is present, contrasting with constitutive genes that are almost always active. Beyond these intricate protein-DNA interactions, gene expression can also be regulated by epigenetic changes. These are modifications to the chromatin, the complex of DNA and protein in eukaryotic cells, that don't alter the underlying DNA sequence, but can profoundly affect how accessible genes are for transcription. It's like adjusting the lighting and acoustics of the stage, setting the mood for which parts of the symphony can be heard most clearly. And so, we arrive at the profound connection between genes, development, and evolution. Development is the remarkable process by which a multicellular organism, starting from a single fertilized egg, undergoes a series of transformations, taking on the various characteristic forms of its life cycle. This astonishing journey is underpinned by four key processes: determination, differentiation, morphogenesis, and growth. Determination sets the developmental fate of a cell, gradually narrowing its potential. Differentiation is the process by which specialized cells, muscle cells, nerve cells, skin cells, arise from less specialized ones, like stem cells. Stem cells, those undifferentiated or partially differentiated cells, are the true marvels of development, capable of becoming many different cell types and proliferating indefinitely. The astounding thing is that, with very few exceptions, cellular differentiation almost never involves a change in the DNA sequence itself. Every cell in your body, from your brain to your bone, contains the same genetic instructions. What makes them different is which genes are expressed and to what extent. A masterclass in gene regulation and epigenetics. Morphogenesis, the development of the organism's body form, is the direct result of spatial differences in gene expression. Imagine a sculptor carefully shaping clay, but instead of hands, the sculptor is a network of genes precisely activated in different regions of the developing embryo. A small, yet incredibly powerful, fraction of the organism's genome forms what biologists call the developmental genetic toolkit. These toolkit genes are ancient, highly conserved across vast evolutionary distances, meaning they're remarkably similar in widely separated groups of animals, from insects to humans. Slight differences in how these toolkit genes are deployed, when they are switched on or off, where they are expressed, and for how long, can lead to dramatic differences in body plan, the number and identity of body parts. Among the most important of these toolkit genes are the Hox genes. These genes act like master architects, determining where repeating parts, such as the many vertebrae of a snake or the segments of an insect, will grow in a developing embryo or larva. The precise sequence and expression patterns of Hox genes dictate the anterior-posterior axis, effectively telling different parts of the embryo what to become and where. It's a staggering testament to the power of a few key genetic instructions to sculpt the vast diversity of life we see around us. This genetic symphony, with its intricate mechanisms of inheritance, expression, and regulation, is not merely the story of how life functions, but how it has evolved, how it continues to adapt, and how the incredible variety of forms and functions on our planet came to be. It is a story told in the language of molecules, yet it speaks to the profound beauty and ingenuity of life itself. And as we conclude this segment, we are left with the undeniable truth that within the double helix of DNA lies the code, the instructions for every living thing, past, present, and future, continuing to compose the grand tapestry of life on Earth. From the quiet hum of a single cell to the sprawling complexity of an entire rainforest, life on Earth is a grand, continuous performance. And at the heart of this unfolding drama lies a story of transformation, adaptation, and an enduring dance with time itself. We are about to delve into the very essence of this story, exploring how life, in all its astonishing forms, has woven itself into the fabric of our planet over billions of years. Think for a moment about the concept of time in a biological sense. It's not just the tick-tock of a clock, but a vast, unimaginable canvas upon which countless generations have played out their existence. This isn't merely about the progression of events. It's about the underlying mechanisms that have allowed life to persist, diversify, and continually redefine itself in the face of an ever-changing world. We're talking about evolution, the central, unifying concept that explains the incredible biological diversity we see around us. The earliest whispers of scientific understanding, including the foundations of what we now call biology, can be traced back to the ancient civilizations of Egypt and Mesopotamia, thousands of years before the Common Era. Their observations, their attempts to understand the natural world and the human body, laid crucial groundwork. But it was in ancient Greece, with thinkers like Aristotle, that a more systematic approach began to emerge. Aristotle, often considered one of the fathers of biology, delved deep into the exploration of biological causation, the why and how of life, and meticulously documented the astounding diversity of organisms he encountered. His successor, Theophrastus, then pioneered the scientific study of plants, beginning to categorize and understand their forms and functions. Fast forward through the centuries, and we find the rich scholarship of the medieval Islamic world, where figures like Al-Jaiz explored zoology, Al-Dinawari wrote extensively on botany, and Rhazes made significant contributions to anatomy and physiology. These scholars, often building upon Greek traditions, meticulously preserved and expanded upon existing knowledge, particularly in the fields of medicine and natural history. Their contributions were vital bridges, carrying scientific understanding forward through periods of immense cultural and intellectual change. But the true acceleration of biological understanding, the moment when we truly began to see the hidden worlds around us, came with a technological marvel, the dramatic improvement of the microscope by Anton van Leeuwenhoek. Suddenly, the invisible became visible. Scholars peered through lenses and discovered entirely new realms of life: spermatozoa, bacteria, infusoria, and an astonishing array of microscopic organisms previously unimagined. This invention was nothing short of revolutionary. It sparked a renewed interest in fields like entomology and spurred the development of advanced techniques for microscopic dissection and staining, fundamentally transforming how we perceived life itself. This era of microscopic revelation profoundly impacted biological thought, leading, in the early 19th century, to the profound realization of the central importance of the cell. In 1838, Matthias Schleiden and Theodor Schwann began to champion the ideas that cells are the basic units of all organisms, and that individual cells possess all the characteristics of life. While they initially clung to the idea of spontaneous generation for new cells, Robert Remak and Rudolf Virchow later solidified the third, crucial tenet, that all cells arise from the division of other existing cells. By the 1860s, these three principles had converged into what we now know as the universal cell theory, a cornerstone of modern biology. While microscopes unveiled the microcosmic, natural historians were busy mapping the macroscopic world. Taxonomy and classification became paramount. Carl Linnaeus, in 1735, published a foundational taxonomy, and by the 1750s, introduced the binomial nomenclature system, those familiar two-part scientific names we use for species today. Around the same time, Georges-Louis Leclerc, Comte de Buffon, was challenging conventional wisdom, suggesting that species were not fixed entities but malleable forms, even hinting at the audacious possibility of common descent, the idea that all life might share an ancestry. These early hints of evolutionary thinking began to coalesce in a serious scientific inquiry with the work of Jean-Baptiste Lamarck. He presented a coherent theory of evolution, proposing that acquired characteristics could be inherited. While his proposed mechanism wasn't entirely correct, his work was a critical step in placing the concept of evolution firmly within scientific discourse. But it was the British naturalist Charles Darwin who, in the mid-19th century, synthesized a truly robust and revolutionary theory. Drawing upon his extensive observations during the Beagle voyage, his deep morphological expertise, the biogeographical insights of Humboldt, the uniformitarian geology of Lyell, and even Malthus's writings on population growth, Darwin forged the theory of evolution by natural selection. Simultaneously and independently, Alfred Russel Wallace arrived at remarkably similar conclusions, a testament to the compelling nature of the evidence. Darwin's monumental work proposed that individuals with heritable traits better suited to their environment are more likely to survive and reproduce, leading to the gradual accumulation of these favorable traits over successive generations, thereby refining the exquisite match between organisms and their environments. This was a paradigm shift, reshaping our understanding of life's history and its ongoing dynamic. Yet, even as Darwin's theory took hold, the precise mechanisms of inheritance remained a mystery. That mystery began to unravel with the quiet, meticulous work of an Austrian monk named Gregor Mendel. In 1865, Mendel published his groundbreaking findings on pea plants, outlining the fundamental principles of biological inheritance. The idea of discrete genetic characteristics, or alleles, passed from parent to offspring. The concepts of dominance and recessiveness, the segregation of alleles during gamete formation, and the independent assortment of different traits. The significance of Mendel's work, however, was not widely recognized until the early 20th century. When it was rediscovered, it provided the missing piece that allowed Darwinian evolution and classical genetics to be reconciled, leading to the modern synthesis, a unified theory that remains the bedrock of evolutionary biology today. The middle of the 20th century brought another seismic shift. In the 1940s and early 1950s, a series of elegant experiments, most famously by Alfred Hershey and Martha Chase, demonstrated conclusively that DNA, not protein, was the component of chromosomes responsible for carrying genetic information. This pivotal discovery paved the way for the ultimate revelation. In 1953, James Watson and Francis Crick, building on the work of Rosalind Franklin and Maurice Wilkins, unveiled the double helical structure of DNA. This discovery marked the true transition to the era of molecular genetics, fundamentally transforming our understanding of life at its most basic level. From the 1950s onward, biology has expanded exponentially into the molecular domain. The genetic code itself, the language by which DNA's information is translated into proteins, was deciphered by scientists like Hargobind Khorana, Robert W. Hawley, and Marshall Warren Nirenberg. This profound understanding of DNA's structure and function culminated in ambitious projects like the Human Genome Project, launched in 1990, which set out to map the entire human genetic blueprint. This journey, from ancient observations to the deepest molecular secrets of life, represents a stunning testament to human curiosity and ingenuity, continually revealing the intricate, grand tapestry of time that is the history of life on Earth. From the very smallest units of life to the vast, intricate webs that span our entire planet, the story of biology is one of endless connection. It's about how everything, from a single-celled microbe to the largest redwood, exists within a grand, interconnected system. Today, we delve into these interwoven worlds, exploring Earth's incredible biodiversity, the complex dance of ecosystems, and why the imperative of conservation has become perhaps the most critical challenge of our time. Biology, at its heart, is the scientific pursuit of understanding life itself. It's a sweeping natural science that touches upon everything from the structure of a protein to the evolution of a species, from the flow of energy in a single cell to the distribution of entire ecosystems. 5 fundamental themes continually emerge as we explore this vast subject. The cell as life's basic unit, the blueprint of genes and inheritance, the ceaseless engine of evolution driving diversity, the critical transformation of energy that sustains all living processes, and a remarkable ability of organisms to maintain internal stability, a concept we call homeostasis. We examine life across an astonishing spectrum of organization. It begins with the infinitesimal, molecules and cells, and scales up through individual organisms, vast populations, and ultimately, to the planetary tapestry of ecosystems. This incredible breadth has given rise to a myriad of specialized fields. Molecular biology, peering into the very machinery of life. Physiology, deciphering how bodies function. Ecology, revealing the intricate relationships within nature. Evolutionary biology, charting life's epic journey through time. Developmental biology, observing how complexity unfolds from a single cell. And systematics, the science of classifying and naming life. Each of these disciplines employs a unique blend of observation, rigorous experimentation, and mathematical modeling to unlock the secrets of biological phenomena. Modern biology, in its very essence, stands firmly on two monumental pillars. The first is Charles Darwin's theory of evolution by natural selection, which provided the unifying framework for understanding the stunning diversity of life. The second is our molecular comprehension of genes, intricately encoded within the double helix of DNA. The groundbreaking discovery of DNA's structure and the subsequent explosion of molecular genetics have profoundly reshaped nearly every corner of biology, leading to transformative applications in medicine, agriculture, biotechnology, and environmental science. It's truly astounding to consider that life on Earth is believed to have first sparked into existence over 3.7 billion years ago. From those primordial beginnings, it has diversified into an almost unimaginable array of organisms. From the simplest single-celled archaea and bacteria, resilient survivors from the dawn of time, to the breathtaking complexity of multicellular plants, fungi, and animals. Biologists tirelessly work to classify these myriad forms, tracing their shared characteristics and evolutionary relationships through meticulous taxonomic and phylogenetic frameworks. Within ecosystems, these organisms engage in a continuous, dynamic interplay, driving the essential cycles of energy flow and nutrient circulation that sustain the entire planetary system. Biology remains a field of constant discovery, perpetually integrating new technologies and insights. It's a science that not only deepens our understanding of life and its fundamental processes, but also actively contributes to finding solutions for some of humanity's most pressing challenges, from battling disease and mitigating climate change to safeguarding the irreplaceable tapestry of biodiversity. The very word biology itself offers a glimpse into its foundational meaning. It stems from the Greek bios, meaning life, and logia, meaning the study of. The compound term, in its modern sense, began to emerge independently in the early 19th century, notably articulated by Gottfried Reinhold Treviranus in his 1802 work, Biologie oder Philosophie der lebenden Natur, and by Jean-Baptiste Lamarck. These pioneers were giving a name to a burgeoning field that sought to unify the study of all living things. The earliest glimmers of scientific inquiry, including early forms of medicine, can be traced back to the ancient civilizations of Egypt and Mesopotamia, roughly between 300 and 1200 BCE. Their foundational contributions paved the way for ancient Greek natural philosophy. Philosophers like Aristotle, living from 384 to 322 BCE, made extensive contributions, exploring biological causation and meticulously documenting the diversity of life he observed. His successor, Theophrastus, initiated the scientific study of plants. Later, during the medieval Islamic world, scholars like Al-Jaiz, Al-Danawari, who wrote extensively on botany, and Rhazes, who delved into anatomy and physiology, further advanced biological knowledge, often building upon the traditions of Greek philosophy. Biology truly began to accelerate with the dramatic improvements to the microscope by Anton van Leeuwenhoek. It was through his lenses that scholars first glimpsed the unseen world: spermatozoa, bacteria, infusoria, and an astonishing diversity of microscopic life. Jan Swammerdam's meticulous investigations sparked a renewed interest in entomology and helped refine techniques of microscopic dissection and staining, forever changing how we viewed the intricate structures of living things. These advancements in microscopy had a profound impact on biological thought. By the early 19th century, the central importance of the cell was coming into sharp focus. In 1838, Matthias Schleiden and Theodor Schwann began to champion the revolutionary and now universally accepted ideas that the cell is the basic unit of all organisms and that individual cells possess all the characteristics of life. Initially, they resisted the third crucial tenet, that all cells arise from the division of other cells, still clinging to notions of spontaneous generation. However, Robert Remak and Rudolf Virchow eventually provided the irrefutable evidence for this third principle, and by the 1860s, cell theory, as we know it today, was firmly established. Simultaneously, natural historians were dedicating themselves to the monumental task of taxonomy and classification. Carl Linnaeus, in 1735, published a foundational taxonomy for the natural world, and in the 1750s, introduced the binomial nomenclature system that gives scientific names to all species. Georges-Louis Leclerc, Comte de Buffon, began to challenge the idea of fixed species, suggesting that living forms were malleable and even hinting at the possibility of common descent, a truly radical idea for his time. Serious evolutionary thinking gained significant traction with the works of Jean-Baptiste Lamarck, who put forth a coherent theory of evolution, though his proposed mechanism of acquired characteristics would later be disproven. However, it was the British naturalist Charles Darwin, drawing upon the biogeographical insights of Alexander von Humboldt, the uniformitarian geology of Charles Lyell, Thomas Malthus's writings on population growth, and his own profound morphological expertise and extensive natural observations, who forged a far more successful and enduring evolutionary theory based on natural selection. Remarkably, Alfred Russel Wallace, working independently, arrived at strikingly similar conclusions with compelling evidence. The very foundation of modern genetics was laid by the work of Gregor Mendel in 1865. His meticulous experiments with pea plants outlined the fundamental principles of biological inheritance, though the profound significance of his work remained largely unrecognized until the early 20th century. It was then that a modern synthesis emerged, reconciling Darwinian evolution with classical genetics, unifying these two powerful ideas into a cohesive theory. In the 1940s and early 1950s, a series of pivotal experiments, most notably by Alfred Hershey and Martha Chase, unequivocally pointed to DNA as the molecule responsible for carrying genetic traits. The focus then shifted to new kinds of model organisms, such as viruses and bacteria, which proved instrumental in unlocking further secrets. This era culminated in the groundbreaking discovery of the double helical structure of DNA by James Watson and Francis Crick in 1953, marking the definitive transition into the age of molecular genetics. From the 1950s onward, biology has expanded exponentially into the molecular domain. The genetic code itself was cracked by Har Gobind Khorana, Robert W. Hawley, and Marshall Warren Nirenberg, once the concept of DNA codons was understood. This relentless pursuit of knowledge reached another landmark with the launch of the Human Genome Project in 1990, a monumental endeavor to map the entire human genetic blueprint. At its core, all life is fundamentally built upon chemical elements. Oxygen, carbon, hydrogen, and nitrogen collectively account for a staggering 96% of the mass of all organisms. The remaining essential components primarily consist of calcium, phosphorus, sulfur, sodium, chlorine, and magnesium. These elements, in various combinations, form compounds crucial for life, such as water. Biochemistry is the study that delves into these intricate chemical processes within and related to living organisms. Molecular biology, a branch of this field, aims to understand the molecular basis of biological activity within and between cells, including the sophisticated processes of molecular synthesis, modification, mechanisms, and interactions. It is a profound thought that life itself arose from Earth's primordial ocean, forming some 3.8 billion years ago. To this day, water remains the most abundant and indispensable molecule in every living organism. Its importance cannot be overstated. Water's effectiveness as a solvent is key. It can dissolve a multitude of solutes, such as sodium and chloride ions, or countless other small molecules to form an aqueous solution. Once dissolved in water, these solutes are far more likely to encounter one another and thus participate in the myriad chemical reactions that sustain life. From a molecular perspective, water is a small, polar molecule with a distinctive bent shape. This shape is created by the polar covalent bonds between two hydrogen atoms and one oxygen atom, H2O. Because the OH bonds are polar, the oxygen atom carries a slight negative charge, while the two hydrogen atoms each possess a slight positive charge. This inherent polarity allows water molecules to attract each other through hydrogen bonds, making water remarkably cohesive. This cohesive force, especially at the surface of a liquid, gives rise to surface tension. Water is also adhesive, meaning it can cling to the surfaces of other polar or charged non-water molecules. One of water's truly unique properties is that it is denser as a liquid than it is as a solid or ice. This remarkable characteristic is why ice floats on liquid water in ponds, lakes, and oceans. This floating ice acts as an insulator, protecting the liquid water below from freezing solid and cold air, thereby allowing aquatic life to persist through harsh winters. Furthermore, water possesses a high specific heat capacity, meaning it can absorb a substantial amount of energy before its temperature significantly increases, far more than other common solvents like ethanol. Consequently, a large amount of energy is required to break the hydrogen bonds between water molecules to transform liquid water into water vapor, a property vital for temperature regulation in organisms. Even as a molecule, water is not entirely stable. Each water molecule continually dissociates into hydrogen and hydroxyl ions before swiftly reforming. In pure water, the balance between hydrogen and hydroxyl ions is equal, resulting in a neutral pH. Moving beyond water, we encounter organic compounds, the very backbone of life. These are molecules characterized by carbon atoms bonded to other elements, most notably hydrogen. With the sole exception of water, nearly every molecule that constitutes an organism contains carbon. Carbon's extraordinary ability to form covalent bonds with up to 4 other atoms allows it to construct an astonishing diversity of large, complex molecules. For instance, a single carbon atom can form 4 single covalent bonds, as seen in methane, or 2 double covalent bonds, as in carbon dioxide, or even a triple covalent bond, as in carbon monoxide. Moreover, carbon can link together to form incredibly long chains of carbon-carbon bonds, like an octane, or intricate ring-like structures, such as glucose. The simplest organic molecules are hydrocarbons, a large family composed solely of hydrogen atoms bonded to a chain of carbon atoms. This hydrocarbon backbone can then be adorned with other elements like oxygen, phosphorus, and sulfur, which dramatically alter the chemical behavior of the compound. These groups of atoms, containing elements like O, H, P, and S, bonded to a central carbon atom or skeleton, are known as functional groups. There are 6 prominent functional groups found in organisms: the amino group, carboxyl group, carbonyl group, hydroxyl group, phosphate group, and sulfhydryl group, each imparting distinct chemical properties. A pivotal experiment in 1953, the Miller-Urey experiment, famously demonstrated that organic compounds could be synthesized abiotically within a closed system designed to mimic the conditions of early Earth. This groundbreaking work strongly suggested that complex organic molecules, the precursors to life, could have spontaneously arisen on our young planet, laying the groundwork for what we call abiogenesis. These smaller organic compounds then assemble into larger structures known as macromolecules. These are vast molecules, built from smaller repeating subunits called monomers. Monomers include the fundamental building blocks of life: sugars, amino acids, and nucleotides. Carbohydrates, for instance, encompass both individual sugar monomers and their complex polymer chains. Lipids are a unique class of macromolecules because they are not formed from repeating polymer subunits. This diverse group includes steroids, phospholipids, and fats, all characterized by their largely nonpolar and hydrophobic, or water-repelling, nature. Proteins are arguably the most diverse of all macromolecules, performing an astounding array of functions. They encompass enzymes, the catalysts of life, transport proteins, moving substances across membranes, large signaling molecules, mediating cellular communication, antibodies, defending against invaders, and structural proteins, providing shape and support. The basic unit, or monomer, of a protein is an amino acid, and remarkably, only 20 different amino acids are utilized to construct the vast multitude of proteins found in life. Finally, we have nucleic acids, the polymers of nucleotides. Their monumental function is to store, transmit, and express hereditary information, the very blueprint of life. At the heart of every living thing, from the simplest bacterium to the most complex human, lies the cell. Cell theory, a cornerstone of biology, states that cells are the fundamental units of life, that all living things are composed of one or more cells, and crucially, that all cells arise from pre-existing cells through cell division. Most cells are incredibly small, typically ranging from 1 to 100 micrometers in diameter, making them visible only under the magnification of a light or electron microscope. Broadly, there are two main types of cells: eukaryotic cells, which possess a distinct nucleus housing their genetic material, and prokaryotic cells, which lack a nucleus. Prokaryotes, such as bacteria, are always single-celled organisms, while eukaryotes can be either single-celled, like many protists, or multicellular, forming the complex tissues and organs of plants, fungi, and animals. In multicellular organisms, every single cell in the body ultimately traces its lineage back to a single, fertilized egg cell. Let's delve into the intricate structure of cell. Every cell is enveloped by a cell membrane, a delicate yet crucial barrier that separates its internal contents, the cytoplasm, from the external extracellular space. This cell membrane is composed of a lipid bilayer, a double layer of phospholipid molecules. Embedded within this bilayer, cholesterol molecules help maintain its fluidity across a range of temperatures. The cell membrane is semi-permeable, selectively allowing small molecules like oxygen, carbon dioxide, and water to pass through, while restricting the movement of larger molecules and charged particles, such as ions. Within the cell membrane are also numerous membrane proteins. Integral membrane proteins span the entire membrane, serving as transporters, channels, or receptors. Peripheral proteins, on the other hand, attach more loosely to the outer or inner surface of the membrane, acting as enzymes or shaping the cell. Cell membranes are integral to a variety of vital cellular processes, including cell adhesion, the storage of electrical energy, and cell signaling, and they provide the attachment points for various extracellular structures like cell walls, glycocalyxes, and the internal cytoskeleton. Inside the cytoplasm of a cell, a rich soup of biomolecules thrives, including countless proteins and nucleic acids. Eukaryotic cells, in particular, boast specialized structures called organelles, many of which are themselves enclosed within their own lipid bilayers or exist as distinct spatial units. These organelles include the cell nucleus, the command center containing most of the cell's DNA, and mitochondria, the powerhouses that generate adenosine triphosphate, or ADP, to fuel all cellular processes. Other organelles, such as the endoplasmic reticulum and Golgi apparatus, play crucial roles in the synthesis and packaging of proteins, respectively. Biomolecules, including waste proteins, can be broken down and recycled within lysosomes, another specialized organelle. Plant cells exhibit additional organelles that distinguish them from animal cells. A robust cell wall provides structural support, while chloroplasts are the sites of photosynthesis, harvesting sunlight energy to produce sugars. Large vacuoles provide storage, structural support, and are involved in plant reproduction and seed breakdown. Eukaryotic cells also possess a dynamic cytoskeleton, a network of microtubules, intermediate filaments, and microfilaments. This internal scaffolding not only provides structural support, but is also essential for cell movement and the precise movement of organelles within the cell. Microtubules are built from tubulin proteins, intermediate filaments from various fibrous proteins, and microfilament from actin molecules, which interact with other protein strands to generate movement. All cells, without exception, require energy to sustain their myriad processes. Metabolism is the collective term for the entire set of chemical reactions occurring within an organism. The three primary purposes of metabolism are converting food into usable energy to power cellular activities, transforming food or fuel into the monomer building blocks needed to construct larger molecules, and efficiently eliminating metabolic waste products. These enzyme-catalyzed reactions are what enable organisms to grow, reproduce, maintain their complex structures, and respond to their ever-changing environments. Metabolic reactions can be broadly categorized into two types. Catabolic reactions involve the breaking down of complex compounds, such as the breakdown of glucose into pyruvate during cellular respiration, typically releasing energy in the process. Anabolic reactions, conversely, involve the building up or synthesis of complex compounds like proteins, carbohydrates, lipids, and nucleic acids, and these processes usually consume energy. The chemical reactions of metabolism are meticulously organized into metabolic pathways, where one chemical is progressively transformed through a series of steps into another, with each step typically facilitated by a specific enzyme. Enzymes are absolutely crucial to metabolism. They act as biological catalysts, allowing reactions that require energy to proceed rapidly by coupling them to spontaneous reactions that release energy. Enzymes dramatically reduce the activation energy needed to convert reactants into products, speeding up reactions without being consumed in the process. They also provide a critical mechanism for regulating the rate of metabolic reactions, allowing cells to respond dynamically to changes in their environment or to signals from other cells. Cellular respiration is a fundamental set of metabolic reactions and processes that occur in cells to convert the chemical energy stored in nutrients into adenosine triphosphate, or ADP, the universal energy currency of the cell, while simultaneously releasing waste products. The reactions involved in respiration are primarily catabolic, meaning they break down large molecules into smaller ones, releasing energy in the process. Respiration is one of the key ways a cell unlocks chemical energy to fuel all its activities. The overall process unfolds in a series of biochemical steps, many of which involve redox reactions where electrons are transferred. While cellular respiration is technically a combustion reaction, it doesn't resemble a rapid fire in a cell due to the slow, controlled release of energy through these intricate series of reactions. Sugar, primarily in the form of glucose, is the main nutrient utilized by both animal and plant cells in respiration. Cellular respiration that involves oxygen is termed aerobic respiration, and it proceeds through four distinct stages: glycolysis, the citric acid cycle, also known as the Krebs cycle, the electron transport chain, and oxidative phosphorylation. Glycolysis, the initial metabolic process, occurs in the cytoplasm, where glucose is converted into two pyruvate molecules, yielding a net production of two ADP molecules. Each pyruvate is then oxidized into acetyl-CoA by the pyruvate dehydrogenase complex, a reaction that also generates NADH and carbon dioxide. Acetyl-CoA then enters the citric acid cycle, which takes place within the mitochondrial matrix. At the completion of this cycle, the total yield from one glucose molecule, or two pyruvates, is 6 NADH, 2 FADH2, and 2 ADP molecules. Finally, the next critical stage is oxidative phosphorylation, which in eukaryotes occurs on the inner mitochondrial cristae. Oxidative phosphorylation consists of the electron transport chain, a series of 4 protein complexes that sequentially transfer electrons from one complex to another. This electron transfer releases energy from NADH and FADH2, which is harnessed to pump protons, or hydrogen ions, across the inner mitochondrial membrane, a process called chemiosmosis. This pumping generates a proton motive force, a powerful gradient that drives the enzyme ATP synthase to synthesize a large amount of additional ADP by phosphorylating ADP molecules. The journey of these electrons culminates with molecular oxygen, serving as the final electron acceptor. However, if oxygen is not available, pyruvate does not enter cellular respiration. Instead, it undergoes a process called fermentation. In this anaerobic pathway, pyruvate remains in the cytoplasm and is converted into various waste products that can then be removed from the cell. The primary purpose of fermentation is to oxidize the electron carriers, specifically NADH back to NAD+, so that glycolysis can continue to produce a small amount of ADP and to remove the accumulating pyruvate. This waste product varies depending on the organism. In our own skeletal muscles, during strenuous exercise when oxygen demand outstrips supply, the waste product is lactic acid. This is known as lactic acid fermentation. When oxygen becomes available during recovery, NAD+ can then attach to hydrogen from lactate to form ADP. In yeast, the waste products are ethanol and carbon dioxide, a process known as alcoholic or ethanol fermentation. The ADP generated in fermentation is produced solely by substrate-level phosphorylation, a process that does not require oxygen. Moving to the other fundamental energy pathway, photosynthesis is the ingenious process utilized by plants and certain other organisms to convert light energy into chemical energy. This stored chemical energy, typically in carbohydrate molecules like sugars, can then be released later to fuel the organism's metabolic activities through cellular respiration. In most instances, oxygen is released as a vital waste product. The vast majority of plants, algae, and cyanobacteria perform photosynthesis, a process largely responsible for both producing and maintaining the oxygen content of Earth's atmosphere and supplying most of the energy necessary for life on our planet. Photosynthesis unfolds in 4 intricate stages: light absorption, electron transport, ADP synthesis, and carbon fixation. Light absorption is the initial step, where light energy is captured by chlorophyll pigments embedded within proteins in the thylakoid membranes of chloroplasts. This absorbed light energy is then used to remove electrons from a donor molecule, typically water, and transfer them to a primary electron acceptor, a quinone designated as Q. In the second stage, these electrons move from the quinone through a series of electron carriers until they reach a final electron acceptor, which is usually the oxidized form of an ADP+, reducing it to an ADPH. This process largely occurs within a protein complex called photosystem I, or psi. The transport of these electrons is intricately coupled to the movement of protons, or hydrogen ions, from the stroma into the thylakoid lumen, creating a pH gradient across the membrane as hydrogen ions become more concentrated inside the lumen. This process is strikingly analogous to the proton motive force generated across the inner mitochondrial membrane during aerobic respiration. During the third stage of photosynthesis, the flow of protons down their concentration gradient from the thylakoid lumen back to the stroma through the ADP synthase enzyme drives the synthesis of ADP. Both the NADPH and ADP generated by these light-dependent reactions in the second and third stages provide the essential energy and electrons to power the synthesis of glucose. This occurs by fixing atmospheric carbon dioxide into existing organic carbon compounds, such as ribulose bisphosphate, RuBP, in a sequence of light-independent, or dark, reactions collectively known as the Calvin cycle. Beyond the internal workings of a single cell, life also depends on the remarkable ability of cells to communicate with each other and their environment, a process known as cell signaling. Signals can be non-chemical, such as light, electrical impulses, or heat, or more commonly, chemical signals called ligands that interact with specific receptors. These receptors can be embedded in the cell membrane of another cell or located deep inside a cell. Generally, there are 4 types of chemical signals: autocrine, paracrine, juxtacrine, and hormones. In autocrine signaling, the ligand affects the very same cell that releases it. For instance, tumor cells can reproduce uncontrollably because they release signals that trigger their own self-division. In paracrine signaling, the ligand diffuses only to nearby cells and affects them. A classic example is neurons, which release neurotransmitters that diffuse across a synaptic cleft to bind with receptors on an adjacent cell, such as another neuron or a muscle cell. Juxtacrine signaling involves direct physical contact between the signaling and responding cells. Finally, hormones are ligands that travel through the circulatory systems of animals or the vascular systems of plants to reach their distant target cells. Once a ligand binds with a receptor, it can initiate a cascade of events that influences the behavior of the target cell, depending on the type of receptor. For instance, neurotransmitters binding to an inotropic receptor can alter the electrical excitability of a target cell. Other receptor types include protein kinase receptors, such as the receptor for insulin, and G-protein-coupled receptors, whose activation can initiate complex second messenger cascades. The entire process by which a chemical or physical signal is transmitted through a cell as a series of molecular events is known as signal transduction. The continuation of life, both for individual organisms and for entire lineages, hinges on the cell cycle, a precisely orchestrated series of events that culminates in a cell dividing into two daughter cells. These events include the meticulous duplication of its DNA and some of its organelles, followed by the careful partitioning of its cytoplasm into the two new daughter cells, a process termed cell division. In eukaryotes, that is animal, plant, fungal, and protist cells, there are two distinct and vital types of cell division, mitosis and meiosis. Mitosis is a critical part of the cell cycle, where replicated chromosomes are accurately separated into two new nuclei. This process gives rise to genetically identical cells, where the total number of chromosomes is maintained. Generally, mitosis, the division of a nucleus, is preceded by the S stage of interphase, during which the DNA is replicated and is often followed by telophase and cytokinesis. Cytokinesis is the physical division of the cytoplasm, organelles, and cell membrane, resulting in two new cells, each receiving roughly equal shares of these cellular components. All the stages of mitosis collectively define the mitotic phase of an animal cell cycle, resulting in the division of the mother cell into two genetically identical daughter cells. The cell cycle, driven by mitosis, is a fundamental process by which a single-celled fertilized egg develops into a complex, mature organism. It's also the ongoing process by which tissues like hair, skin, and blood cells are continuously renewed, and some internal organs are repaired. After cell division, each daughter cell then embarks on the interphase of a new cycle. In contrast to mitosis, meiosis is a specialized form of cell division that results in 4 haploid daughter cells. This is achieved through one round of DNA replication, followed by 2 distinct divisions. In the first meiotic division, meiosis I, homologous chromosomes are separated. In the second meiotic division, meiosis II, sister chromatids are separated. Both mitosis and meiosis are essential for sexual reproduction at different points in an organism's life cycle, and both are believed to have been present in the last eukaryotic common ancestor. Prokaryotes, including archaea and bacteria, also undergo cell division, but through a simpler process called binary fission. Unlike the complex spindle apparatus formed during eukaryotic mitosis and meiosis, binary fission in prokaryotes proceeds without it. Before binary fission, the bacterium's DNA, usually tightly coiled, uncoils in duplicates. As the bacterium increases in size, preparing to split, the duplicated DNA is pulled to separate poles of the cell. The growth of a new cell wall, or septum, begins to separate the bacterium, a process triggered by FTSE protein polymerization and the formation of a zooring. This new cell wall fully develops, leading to the complete split of the bacterium into 2 new daughter cells, each containing its own tightly coiled DNA, ribosomes, and plasmids. Meiosis is a central and defining feature of sexual reproduction in eukaryotes. Its most fundamental function appears to be the conservation of the integrity of the genome that is passed on from parents to offspring. Two critical aspects of sexual reproduction, meiotic recombination and outcrossing, are likely maintained due to their adaptive advantages. Meiotic recombination facilitates the repair of genomic DNA damage, while outcrossing, or cross-fertilization, provides genetic complementation, which effectively masks the expression of deleterious recessive mutations. The beneficial effect of genetic complementation, derived from outcrossing, is also referred to as hybrid vigor or heterosis. Charles Darwin, in his 1878 book, The Effects of Cross and Self-Fertilization in the Vegetable Kingdom, eloquently noted at the beginning of Chapter 12 that the first and most important of the conclusions which may be drawn from the observations given in this volume is that generally cross-fertilization is beneficial and self-fertilization often injurious, at least with the plants on which I experimented. Genetic variation, often a byproduct of sexual reproduction, can confer long-term advantages to those sexual lineages that engage in outcrossing, allowing them to adapt and thrive in changing environments. And then we arrive at the very essence of how life perpetuates itself. Genetics, the scientific study of inheritance. Mendelian inheritance, specifically, describes the elegant process by which genes and traits are passed down from parents to their offspring. It's governed by several fundamental principles. First, genetic characteristics, which we call alleles, are discrete units and exist in alternate forms. Think of purple versus white flower color, or tall versus dwarf plant height. Each of these alleles is inherited from one of the two parents. According to the law of dominance and uniformity, some alleles are dominant while others are recessive. An organism possessing at least one dominant allele will display the phenotype or observable trait associated with that dominant allele. During the formation of gametes, or sex cells, the alleles for each gene segregate, ensuring that each gamete carries only one allele for each gene. Heterozygous individuals, those with two different alleles for a trait, will produce gametes with an equal frequency of both alleles. Finally, the law of independent assortment states that genes for different traits can segregate independently during gamete formation, meaning they are unlinked. An important exception to this rule will be traits that are sex-linked, located on the sex chromosomes. Test crosses can be performed to experimentally determine the underlying genotype of an organism exhibiting a dominant phenotype. And the venerable Punnett square remains a valuable tool to predict the results of such genetic crosses. The chromosome theory of inheritance, which posits that genes are physically located on chromosomes, was powerfully supported by Thomas Morgan's experiments with fruit flies, establishing the concept of sex linkage between eye color and sex in these insects. At the heart of inheritance lies the gene, a fundamental unit of heredity. A gene corresponds to a specific region of deoxyribonucleic acid, or DNA, that carries the genetic information controlling the form or function of an organism. DNA itself is an awe-inspiring molecule, composed of two polynucleotide chains that elegantly coil around each other to form the iconic double helix. In eukaryotes, DNA is organized into linear chromosomes, primarily residing in the cell nucleus. In prokaryotes, the DNA typically exists as a circular chromosome, held within a region called the nucleoid. The complete set of chromosomes within a cell is collectively known as its genome, and the full assemblage of genes in an organism constitutes its genotype. DNA replication is a remarkable semi-conservative process, meaning that during duplication, each original strand of the double helix serves as a template for the synthesis of a new, complementary strand of DNA. This ensures faithful transmission of genetic information. However, sometimes errors occur. Mutations are heritable changes in DNA. They can arise spontaneously as a result of replication errors that were not corrected by the cell's proofreading mechanisms, or they can be induced by environmental mutagens. Agents like certain chemicals, such as nitrous acid or benzopyrene, or various forms of radiation, including X-rays, gamma rays, ultraviolet radiation, and particles emitted by unstable isotopes. Mutations can lead to a range of phenotypic effects, from loss of function, where a gene's product no longer works, to gain of function, where it acquires a new activity, or conditional mutations, which only manifest under specific environmental conditions. It's important to remember that not all mutations are detrimental. Some can be beneficial, providing a crucial source of genetic variation that fuels the engine of evolution, allowing populations to adapt and change over time. Others, however, can be harmful, particularly if they result in the loss of function of genes essential for survival. The bridge between a gene, encoded in DNA, and an observable trait, manifest in an organism's physical characteristics, is called gene expression. This is the molecular process by which a genotype gives rise to a phenotype through the production of proteins. This fundamental flow of genetic information is famously summarized by the central dogma of molecular biology, formulated by Francis Crick in 1958. Genetic information flows from DNA to RNA to protein. This occurs in two primary gene expression processes: transcription, where the information from DNA is copied into RNA, and translation, where the RNA sequence is used to synthesize a protein. The precise control of gene expression is paramount for an organism's development and its ability to respond to environmental cues. This regulation can occur at virtually every step of the process. During transcription, when RNA is synthesized from DNA, during RNA splicing, where non-coding regions are removed from RNA, during translation, when RNA is used to build proteins, and even through post-translational modification of the protein itself. Gene expression can be influenced by either positive or negative regulation, depending on how specific regulatory proteins, called transcription factors, bind to DNA sequences near or at a gene's promoter. In prokaryotes and some lower eukaryotes, a cluster of genes that share the same promoter is known as an operon. In positive regulation, an activator protein, a type of transcription factor, stimulates transcription when it binds to a sequence near or at the promoter. Negative regulation, conversely, occurs when another transcription factor, called a repressor, binds to a DNA sequence known as an operator, which is part of an operon, effectively blocking transcription. Repressors can sometimes be inhibited by specific compounds called inducers, such as allolactose, thereby allowing transcription to proceed. Genes that can be activated by inducers are termed inducible genes, in contrast to constitutive genes that are almost constantly active. Separately, structural genes encode proteins that are not directly involved in gene regulation but perform other cellular functions. Beyond these regulatory events involving promoters, gene expression can also be profoundly influenced by epigenetic changes to chromatin, the complex of DNA and protein found in eukaryotic cells, which can alter gene accessibility without changing the underlying DNA sequence. The journey from a single cell to a complex organism is a marvel of biological engineering, driven by genes, development, and evolution. Development is the intricate process by which a multicellular organism, be it a plant or an animal, undergoes a series of profound changes, starting from a single cell and progressively taking on the various forms characteristic of its life cycle. Four key processes underlie this astonishing transformation: determination, differentiation, morphogenesis, and growth. Determination sets the developmental fate of a cell, a fate that becomes progressively more restrictive as development proceeds. Differentiation is the process by which specialized cells, neurons, muscle cells, skin cells, arise from less specialized cells, such as stem cells. Stem cells are remarkable. They are undifferentiated or only partially differentiated cells that possess the ability to differentiate into various types of specialized cells and can proliferate indefinitely to produce more of themselves. Cellular differentiation dramatically alters a cell's size, shape, membrane potential, metabolic activity, and its responsiveness to signals. These profound changes are largely due to highly controlled modifications in gene expression and epigenetics, rather than changes in the DNA sequence itself. Thus, despite having the same genome, Different cells within an organism can exhibit vastly different physical characteristics and functions. Morphogenesis, the development of body form, is the direct result of spatial differences in gene expression. A small but incredibly powerful fraction of the genes in an organism's genome, often referred to as the developmental genetic toolkit, controls the development of that organism. These toolkit genes are remarkably highly conserved across vast evolutionary distances, meaning they are ancient and very similar in widely separated groups of animals, highlighting their fundamental importance. Subtle differences in how these toolkit genes are deployed, when they're turned on, where, and for how long, can profoundly affect the body plan and the number, identity, and pattern of body parts. Among the most crucial of these toolkit genes are the Hox genes, which play a central role in determining where repeating parts, such as the many vertebrae of snakes, will grow in a developing embryo or larva. This brings us to evolution, the central organizing concept in all of biology. Evolution is defined as the change in the heritable characteristics of populations over successive generations. Its profound implications underpin nearly everything we understand about life. Consider the deliberate hand of humans in artificial selection, where animals or plants are selectively bred for specific desirable traits, often over many generations. Darwin observed this and reasoned that, given that traits are inherited, populations naturally contain a varied mix of traits, and that reproduction inevitably leads to an increase in population size. He then inferred that in the natural world, it was nature itself that played the role of the breeder, selecting for specific traits. Darwin famously argued that individuals who possessed heritable traits better adapted to their environments were more likely to survive and, crucially, to produce more offspring than other individuals. He further inferred that this natural selection would lead to the gradual accumulation of favorable traits over successive generations, thereby increasing the match between the organisms and their environment, leading to the astonishing diversity and adaptation we see all around us. The process by which one lineage splits into two, evolving independently from each other, is called speciation. A species, generally, is defined as a group of organisms that can mate with one another and produce fertile offspring. For speciation to occur, a critical prerequisite is reproductive isolation, where populations are prevented from interbreeding. This reproductive isolation can arise from incompatibilities between genes, as described by the Bateson-Dobzhansky-Muller model, where the interaction of genes from different populations leads to reduced fitness in hybrids. Reproductive isolation also tends to increase in direct correlation with genetic divergence between populations. Speciation can occur when there are physical barriers, such as mountains or oceans, that divide an ancestral species, a process known as allopatric speciation. Over vast stretches of time, these geographically separated populations accumulate genetic differences, eventually becoming distinct species. To understand the grand narrative of life, we often rely on phylogeny, the evolutionary history of a specific group of organisms or their genes. This history can be elegantly represented using a phylogenetic tree, a diagram showing lines of descent and relationships among organisms or their genes. Each line drawn on the time axis of such a tree represents a lineage, tracing the descendants of a particular species or population. When a lineage divides into two, it's represented as a fork or split on the tree, symbolizing a speciation event. Phylogenetic trees form the bedrock for comparing and grouping different species. Different species that share a particular feature inherited from a common ancestor are described as having homologous features, or synapomorphies. Phylogeny thus provides the essential basis for biological classification. This system is hierarchical and rank-based, with the highest rank being the domain, followed by kingdom, phylum, class, order, family, genus, and finally, species. All known organisms can be classified into one of three domains: archaea, bacteria, or eukarya, which encompasses the familiar kingdoms of fungi, plants, and animals. The history of life on Earth is an epic saga, tracing how organisms have evolved from the earliest emergence of life to the astounding diversity we witness today. Our planet formed approximately 4.5 billion years ago, and all life on Earth, both living and extinct, is believed to have descended from a single last universal common ancestor that existed about 3.5 billion years ago. Geologists have meticulously developed a geologic timescale, dividing Earth's immense history into major divisions. It begins with 4 eons: the Hadean, Archean, Proterozoic, and Phanerozoic. The first 3 of these are collectively known as the Precambrian, a vast stretch of time lasting approximately 4 billion years. Each eon is further subdivided into eras, with the Phanerozoic eon, which began 539 million years ago, being split into the Paleozoic, Mesozoic, and Cenozoic eras. These 3 eras, in turn, comprise 11 periods from the Cambrian through to the Quaternary. The striking similarities among all known present-day species serve as compelling evidence that they have diverged through the process of evolution from this common ancestor. Biologists universally regard the ubiquity and consistency of the genetic code as irrefutable proof of universal common descent for all bacteria, archaea, and eukaryotes. In the early Archean eon, microbial mats, composed of coexisting bacteria and archaea, with a dominant form of life, and many of the major steps in early evolution are thought to have unfolded within these ancient microbial communities. The earliest definitive evidence of eukaryotes dates back approximately 1.85 billion years ago, and while they may have been present earlier, their diversification dramatically accelerated once they began to utilize oxygen in their metabolism. Later, around 1.7 billion years ago, multicellular organisms began to emerge, with their cells becoming differentiated to perform specialized functions. Algae-like multicellular land plants trace their origins back to about 1 billion years ago, although evidence suggests that microorganisms formed the earliest terrestrial ecosystems far earlier, at least 2.7 billion years ago, paving the way for the eventual colonization of land by plants during the Ordovician period. These early land plants were so successful that their immense growth and subsequent burial are thought to have contributed to the late Devonian extinction event by significantly altering atmospheric CO2 levels. The enigmatic Ediacara biota appeared during the Ediacaran period, preceding the incredible burst of diversification known as the Cambrian explosion, about 525 million years ago, which saw the origin of vertebrates along with most other modern animal phyla. During the Permian period, synapsids, including the ancestors of mammals, dominated the land. However, most of this diverse group perished in the catastrophic Permian Triassic extinction event 252 million years ago, a time often referred to as the Great Dying. In the long recovery from this global catastrophe, archosaurs, a group that includes crocodiles, birds, and dinosaurs, rose to become the most abundant land vertebrates. One archosaur group, the dinosaurs, truly dominated the Jurassic and Cretaceous periods. After the Cretaceous, Paleogene extinction event 66 million years ago, which wiped out the non-avian dinosaurs, mammals rapidly increased in size and diversity, seizing the ecological opportunities left vacant. Such mass extinctions, though devastating, may have accelerated evolution by opening up vast new niches, providing opportunities for new groups of organisms to diversify and thrive. The astonishing diversity of life on Earth is categorized into 3 fundamental domains: bacteria, archaea, and eukaryotes. Bacteria constitute a vast domain of prokaryotic microorganisms, typically a few micrometers in length, exhibiting a variety of shapes from spheres to rods and spirals. They were among the first life forms to grace our planet and are found in almost every conceivable habitat. Bacteria thrive in soil, water, acidic hot springs, radioactive waste, and even deep within the Earth's crust. They also form intricate symbiotic and parasitic relationships with plants and animals. Despite their ubiquity, the vast majority of bacteria remain uncharacterized and only a fraction of bacterial phyla have species that can be grown and studied in the laboratory, highlighting the immense hidden diversity yet to be discovered. Archaea represent the other domain of prokaryotic cells. Initially, they were mistakenly classified as bacteria and called archaebacteria, a term that has now fallen out of use due to our enhanced understanding. Archaeal cells possess unique properties that distinctly separate them from both bacteria and eukaryotes. Archaea are further subdivided into multiple recognized phyla. While generally similar to bacteria in size and shape, some archaea, like the flat, square cells of Haloquadratum walsbii, exhibit truly bizarre morphologies. Despite their morphological resemblance to bacteria, archaea possess genes and several metabolic pathways that are more closely related to those of eukaryotes, particularly for the enzymes involved in transcription and translation. Other aspects of archaeal biochemistry are entirely unique, such as their reliance on ether lipids in their cell membranes, including archaeols. Archaea utilize a wider range of energy sources than eukaryotes, from organic compounds like sugars to ammonia, various metal ions, or even hydrogen gas. Salt-tolerant archaea, known as halorchaea, can even use sunlight as an energy source, and some species of archaea fix carbon, though no known archaea perform both processes like plants and cyanobacteria. Archaea reproduce asexually through binary fission, fragmentation, or budding, and unlike bacteria, no known species of archaea form endospores. The first observed archaea were often extremophiles, thriving in extreme environments like hot springs and salt lakes, where other organisms could not survive. However, the development of improved molecular detection tools has led to the discovery of archaea in almost every habitat on Earth, including soil, oceans, and marshlands. Archaea are particularly numerous in the oceans, where the archaea within plankton may constitute one of the most abundant groups of organisms on the planet. They are a significant component of Earth's life and play vital roles in the microbiota of all organisms, including the human gut, mouth, and skin. Their morphological, metabolic, and geographical diversity allows them to fulfill multiple critical ecological roles: carbon fixation, nitrogen cycling, the turnover of organic compounds, and the maintenance of microbial symbiotic and syntrophic communities. Eukaryotes are hypothesized to have branched off from archaea, a pivotal event that was followed by their endosymbioses with bacteria, a process called symbiogenesis, which gave rise to mitochondria and chloroplasts, both now integral parts of modern eukaryotic cells. The major lineages of eukaryotes diversified in the Precambrian, approximately 1.5 billion years ago, and can be broadly classified into 8 major clades: alveolates, excavates, straminopiles, plants, rhizarians, amoebozoants, fungi, and animals. 5 of these clades are collectively known as protists, a somewhat informal grouping of mostly microscopic eukaryotic organisms that are not plants, fungi, or animals. While protists likely share a common ancestor, they do not form a single, distinct clade, as some protists are more closely related to plants, fungi, or animals than they are to other protists. Like terms such as algae, invertebrates, or protozoans, protist is used more for convenience than as a formal taxonomic group. Most protists are unicellular, often referred to as microbial eukaryotes. Plants are primarily multicellular organisms, predominantly photosynthetic eukaryotes belonging to the kingdom Plantae, a definition that excludes fungi and some algae. Plant cells evolved through the endosymbiosis of a cyanobacterium into an early eukaryote about 1 billion years ago, giving rise to chloroplasts. The first several clades that emerged after this primary endosymbiosis were aquatic. Most of these aquatic photosynthetic eukaryotic organisms are collectively described as algae, another convenient term, as not all algae are closely related. Algae comprise several distinct clades, such as glaucophytes, which are microscopic freshwater algae thought to resemble the early unicellular ancestor of Plantae. Unlike glaucophytes, other algal clades, like red and green algae, are typically multicellular. Green algae, in particular, include 3 major clades: chlorophytes, Coleochaetophytes, and stoneworts. Fungi are eukaryotes that have a unique way of obtaining nutrients. They digest food outside their bodies. They secrete powerful digestive enzymes that break down large food molecules into smaller ones, which they then absorb through their cell membranes. Many fungi are also saprobes, meaning they feed on dead organic matter, making them indispensable decomposers in ecological systems, recycling vital nutrients back into the environment. Animals are multicellular eukaryotes with remarkable characteristics. With very few exceptions, animals consume organic material, breathe oxygen, are capable of movement, can reproduce sexually, and develop from a hollow sphere of cells called the blastula during embryonic development. Over 1.5 million living animal species have been formally described, with roughly 1 million of these being insects, but it is estimated that the total number of animal species globally could exceed 7 million. Animals engage in incredibly complex interactions with each other and their environments, forming intricate food webs that are fundamental to ecosystem function. Finally, we have viruses, enigmatic submicroscopic infectious agents that can only replicate inside the living cells of other organisms. Viruses infect all types of life forms, from animals and plants to microorganisms, including bacteria and archaea. While over 6,000 virus species have been meticulously described, viruses are found in almost every ecosystem on Earth and are believed to be the most numerous type of biological entity. The origins of viruses within the evolutionary history of life remain a fascinating mystery. Some theories suggest they may have evolved from plasmids, small, circular pieces of DNA that can move between cells, while others propose they might have evolved from more complex bacteria. In the grand tapestry of evolution, viruses serve as an important means of horizontal gene transfer, a process that significantly increases genetic diversity in a way analogous to sexual reproduction. Because viruses possess some, but not all, of the characteristics traditionally ascribed to life, they have often been described as organisms at the edge of life, or simply as self-replicators, blurring the lines of what it truly means to be alive. Now, let's turn our attention to ecology, the profound study of the distribution and abundance of life, and the intricate interactions between organisms and their environment. This field helps us understand how living things exist within and shape the world around them. The fundamental unit of study in ecology is the ecosystem, which is defined as a community of living or biotic organisms interacting in conjunction with the non-living or abiotic components of their environment. These abiotic components include crucial elements like water, light, radiation, temperature, humidity, the atmosphere, acidity, and soil. These biotic and abiotic elements are not isolated, but are inextricably linked together through vital nutrient cycles and energy flows. Energy, almost universally, enters the system from the sun through the miraculous process of photosynthesis and is then incorporated into plant tissue. As animals feed on plants and on one another, they act as conduits, moving both matter and energy through the entire system. They also significantly influence the quantity of plant and microbial biomass present. Crucially, decomposers, organisms like bacteria and fungi, break down dead organic matter, releasing essential carbon back into the atmosphere and facilitating nutrient cycling by converting nutrients stored in dead biomass back into forms that can be readily used by plants and other microbes, completing the cycle. Within these ecosystems, we find populations, which are defined as groups of organisms of the same species occupying a particular area and capable of reproducing from generation to generation. The size of a population can be estimated by multiplying its density by the area or volume it occupies. A critical concept in population ecology is the carrying capacity of an environment, which represents the maximum population size of a species that can be sustained indefinitely by that specific environment. This capacity is determined by the availability of essential resources such as food, habitat, water, and other necessities. The carrying capacity of a population is not static. It can be significantly affected by changing environmental conditions, such as fluctuations in resource availability and the metabolic cost of maintaining those resources. In human populations, new technologies like those from the Green Revolution have historically helped increase Earth's carrying capacity for humans over time, a factor that has often stymied the dire predictions of impending population decline, most famously articulated by Thomas Malthus in the 18th century. Beyond individual populations, we observe communities, which are groups of populations of different species occupying the same geographical area at the same time. Within these communities, biological interactions are the effects that pairs of organisms living together have on each other. These interactions can be either intraspecific, occurring between individuals of the same species, or interspecific, occurring between individuals of different species. These effects can be short-term, like the brief act of pollination or the swift strike of a predator, or they could be long-term, such as the enduring relationship of symbiosis. Both types of interactions often profoundly influence the evolution of the species involved. Symbioses encompass a wide spectrum of long-term interactions, ranging from mutualism, where both partners benefit, to competition, where both partners are harmed. Every species participates as either a consumer, a resource, or both in what are called consumer-resource interactions. These interactions form the very core of food chains, or, more realistically, complex food webs. Within any food web, there are distinct trophic levels. At the lowest level are the primary producers, or autotrophs, such as plants and algae, which convert energy and inorganic material into organic compounds. These organic compounds then become the energy source for the rest of the community. At the next level are the heterotrophs, species that obtain energy by consuming organic compounds from other organisms. Heterotrophs that consume plants are called primary consumers, or herbivores. Those that consume herbivores are secondary consumers, or carnivores. And those that eat secondary consumers are tertiary consumers, and so on. Omnivorous heterotrophs are versatile, able to consume at multiple trophic levels. Finally, there are the decomposers, which feed on the waste products or dead bodies of organisms, returning vital nutrients to the ecosystem. On average, the total amount of energy incorporated into the biomass of a trophic level per unit of time is roughly 1/10 of the energy from the trophic level it consumes. This means that a significant amount, approximately 90% of the energy, is lost as waste and dead material utilized by decomposers, and as heat released from metabolic processes, illustrating the fundamental inefficiency of energy transfer between trophic levels. On the grandest scale, we consider the global ecosystem, or biosphere, the entirety of life on Earth and its interactions with the non-living world. In this immense system, matter exists in different interacting compartments, which can be biotic or abiotic and either accessible or inaccessible, depending on their forms and locations. For example, matter within terrestrial autotrophs, plants, is both biotic and accessible to other organisms, while the matter locked within rocks and minerals is abiotic and largely inaccessible. A biogeochemical cycle describes the pathway by which specific elements of matter are turned over or moved through the biotic components, the biosphere, and the abiotic compartments of Earth, the lithosphere, atmosphere, and hydrosphere. We recognize crucial biogeochemical cycles for essential elements like nitrogen, carbon, and, of course, water. This brings us to one of the most pressing fields in modern biology, conservation biology. This is the scientific study dedicated to the conservation of Earth's biodiversity, with the critical aim of protecting species, their habitats, and entire ecosystems from excessive rates of extinction and the erosion of vital biotic interactions. It concerns itself with the complex factors that influence the maintenance, loss, and restoration of biodiversity, and it applies scientific principles to sustain the evolutionary processes that generate genetic, population, species, and ecosystem diversity. The urgency stems from alarming estimates suggesting that up to 50% of all species on our planet could vanish within the next 50 years. This catastrophic loss is not merely an aesthetic concern. It is already contributing to global poverty, starvation, and, profoundly, will fundamentally reset the course of evolution on this planet. Biodiversity directly impacts the functioning of ecosystems, which in turn provide an invaluable array of services upon which all human societies depend, from clean air and water to food security and climate regulation. Conservation biologists tirelessly research and educate on the trends of biodiversity loss, the escalating rates of species extinctions, and the profound negative effects these are having on our collective ability to sustain the well-being of human society. In response to this unfolding biodiversity crisis, organizations and citizens around the world are actively engaged in conservation action plans, directing crucial research, implementing monitoring programs, and developing educational initiatives that address these concerns from local communities to global scales. The fate of Earth's interconnected worlds, and indeed our own, now rests squarely on our ability to understand, appreciate, and protect this magnificent tapestry of life. And so, our profound journey through the vast and wondrous world of biology comes to a close for tonight. We've gazed into the silent dance of molecules, witnessed the tireless work of cells, and traversed the epic timeline of evolution. I hope this deep dive has offered you new insights. If you've enjoyed this exploration, please consider hitting that like button, sharing your thoughts in the comments, and subscribing to Vibing Science for more cosmic and quantum adventures. What scientific mystery should we unravel next? Until then, keep exploring, keep questioning, and keep vibing with the universe.